enrich and enhance our knowledge and experience in the field of life science, especially in the area of biomedical science. Certainly, this two-day international conference on molecular advances in biomedical research gives away emerging developments in this field. World is directed by the Almighty, and without which nothing is happened. God directs us, guides us in our day-to-day activities. Let us thank our God for this wonderful occasion. Praise Him.
இனிதாய் பாடி இறைவா உன் புகழ் புகழ்கிறோம். சங்கமமாகி சபைதனில் வாழும் சரணம் சரணம் இறைவனே. இறைவணக்கம் இனிதாய் பாடி இறைவா உன் புகழ் புகழ்கிறோம். சங்கமமாகி சபைதனில் வாழும் சரணம் சரணம் இறைவனே. உலகாளும் இறைவா எமையாள வருவாய் என் உணர்வோடு நின்று சபைதனில் அமர்வாய். உலகாளும் இறைவா எமையாள வருவாய் என் உணர்வோடு நின்று சபைதனில் அமர்வாய். சர்வமும் நீயே சகலமும் நீயே, சர்வமும் நீயே சகலமும் நீயே. சரணம் சரணம் திருவடி சரணம். பாக்கள் பாடுகிறோம் பரதம் ஆடுகிறோம் பரம்பொருளாகிய பரமனே. சுரமதில் இணைந்து கரம் ஒன்று குவித்து வரம் ஒன்று தருவாய் வேந்தனே. பாக்கள் பாடுகிறோம் பரதம் ஆடுகிறோம் பரம்பொருளாகிய பரமனே. சுரமதில் இணைந்து கரம் ஒன்று குவித்து வரம் ஒன்று தருவாய் வேந்தனே. சாச சாச கரி சகரி சதரி சாரீரி ரீரி கரி சகரி சதரி சா சாச சாச கரி சகரி சதரி சாரீரி ரீரி கரி சகரி சதரி சாசா சரீரி காம பாதரி தரி சா பதசரிக்கை. இறைவணக்கம் இனிதாய் பாடி இறைவா உன் புகழ் புகழ்கிறோம். சங்கமமாகி சபைதனில் வாழும் சரணம் சரணம் இறைவனே. சரணம் சரணம் இறைவனே. சரணம் சரணம் இறைவனே.
This international conference on molecular advances in biomedical research begins with welcome address. I invite Dr. K. Saravanan Sir, Assistant Professor of Zoology, Nehru Memorial College, to welcome up the gathering.
Respected President Sir, respected Principal, respected our Chief Guest, Dr. Puthanampatti Sir, Dr. Emmanuel Sir, Dr. Ramalingam, Dr. Arjunan Sir, student friends, research scholars, faculty of our department, other department, faculty of university, faculty of other colleges, students, good morning to all. I am very happy to stand before you to welcome the gathering. This two-day conference on molecular advances in biomedical research is need of the hour. When I approached our management to conduct this international conference, our president felt very happy and he motivated and encouraged to conduct this conference. With their felicitation and support, we are taking this institution beyond its horizon. At the outset, I welcome our President, Engineer Pon Balasubramaniam, to this conference. Our President has a great interest in activities such as researches, seminars and conference of this kind. He has been constantly encourages us to take activities like this. I feel very happy to welcome our President, Engineer Pon Balasubramaniam, to this conference. I welcome you, sir. On the line of this, our Principal, Dr. A. Pon Periyasamy, not only has the term Pon in his name, but also he has kindness in all his activities. He works towards predetermined targets and he never deviates from it. As a well-wisher, supporter and guide, he motivates us to take up all our academic activities. On behalf of the conference organizing committee and the Department of Zoology, I welcome you, sir. This inaugural session has eminent resource persons, Dr. P. Puthanampatti, Deputy Director, Lee Kong Chen School of Medicine, Nanyang Technological University, Singapore. He is a man of innovative thoughts in the field of medical research and he readily accepted our invitation and our request to deliver inaugural address. On behalf of the conference organizing committee, I welcome you, sir. We have long association with the Department of Animal Science, Bharat University, Tiruchirappalli. and we frequently share our ideas and experiences in the field of animal science. As a part of our academic efforts, we conduct this conference. Dr. Emmanuel Rajan, Professor and Head, Department of Animal Science, BDU, Tiruchirappalli, helped us to collaborate with the department. With his support, we organized this conference. On behalf of the conference committee and on behalf of the department, I welcome you, sir. Dr. G. Arjunan, UGC BSR Faculty Fellow, Department of Animal Science, BDU, Tiruchirappalli, informed and initiated to conduct this conference in association with us. With his support and guidance, we organized this conference. He is serving for this conference right from the start. I welcome you, sir. It is my duty to welcome the resource persons for this two-day conferences. I welcome Dr. Debashish Das, Chief Scientist, Stem Cell Research Lab, Narayana Nethralaya, Bangalore, Dr. Ramasamy Palmurugan, Associate Professor, Department of Radiology, California, Dr. Murugeshwari, Scientist, Narayana Nethralaya, Bangalore, Dr. Ramalingam, Tuberculosis Institute, Chennai, Dr. Anakesan, Material Synthesis and Characterization Laboratory, Institute of Advanced Technology, University of Putra, Malaysia, Dr. Lenin, Bioscience, Clinical Middle East, Dubai. This conference is full of this resource person and it paves ways for pooling their ideas and knowledge, which will be much beneficial to the participants. Once again, I welcome all the resource persons. I have immense pleasure to welcome the faculty members, research scholars, student participation. Certainly, this conference will fetch a substantial amount of new ideas, facts and experiences to enhance their wisdom and knowledge. It gives me pleasure and happy in welcoming all the faculty members, research scholars, students and Department of Animal Science and other Department of Bharathasan University. Welcome you all. Our HOD Madam and Department faculty members, Dr. P. Neil Narayanan, Dr. N. Ramesh, Dr. Bhubadi Raja, Dr. V. Ramesh, Ms. Tangamani, Dr. Renuga, Dr. Revati, Dr. Sudha and Mr. Kalayarasan take frequent efforts of this kind of academic activities for the enhancement of knowledge of the students of life science. They shoulder responsibilities and accountability for the success of all the activities. I welcome all the faculty members of our department for this conference. Welcome. I welcome our librarian and Dr. A. Venkadeshan, IPC coordinator to the conference. I welcome all the heads of the department and faculty members of college to this conference who continuously encourage us to carry out many academic endeavors. Once again, I welcome you all. I welcome all the participants from various departments of college. Last but not least, I welcome all these who directly or indirectly extend their helping hands in organizing this conference in a grand manner. Once again, welcome every one of this. Thank you. I welcome Dr. Sasikumar sir, head of the department, botany and biotechnology. Welcome you, sir. Thank you.
Thank you, sir. The sun gives light to the world every day. Teachers give light to the students in the form of wisdom and knowledge. Yes, this is the time for lighting the lamp. First of all, important aspect as far as the emerging scientific world is concerned. At this juncture, I call upon Dr. G. Arjunan Sir, UGC BSR Faculty Fellow, Department of Animal Science, Bharathasan University, to give away the introduction about this conference. He is a good academician and an active researcher with innovative thoughts, and he has been contributing a lot to the scientific literature to his credit. Over to Dr. Arjunan Sir. Please, sir.
Engineer Balasubramanian, Principal of the college, Dr. Pon Periyasamy, a keynote address of the international conference. So our friend Dr. P. Puthanampatti from MTU, Dr. Emmanuel Rajan, Head of the Department of Animal Science, Bharathasan University, Dr. Ramalingam, one of the invited speaker, Dr. Sandhi, Head of the Department of Zoology, Nehru Memorial College, and faculty members from Zoology as well as other departments, Dr. Sasikumar from Botany, Priscilla from Bishop College, Uma Maheshwari, EVR College, and Dr. Mahesh from our department, and several other faculty members and students, friends and delegates. So I joined with our organizing secretary, my friend Dr. Saravanan, once again welcome you all for the international conference, the Molecular Advances in Biomedical Research, the two-day international conference. I am very happy to be here to tell a few words about the conference. So the conference means we thought to start up this type of conference in this college. I talked with Dr. Saravanan. And actually this was initiated by Saravanan, where we will be able to conduct this type of conference. So experts we need, and especially the foreign experts. So we have some of the experts we know personally, so they are readily accepted to give a keynote address. That is the one of the main part of the international conference. So in that way, so Dr. Padmanabhan will be contacted, and Kanagesan, and a few others, Dr. Paul Murugan from USA, and one Nicoletto from Italy. So a few others we contacted, they were agreed to. Chandramohan from Saudi Arabia. So these were the experts we contacted. They were agreed to come, at least some, a few. And we thought, okay, we will conduct this conference. And this conference is being conducted in several universities and institutes. So this is the usual process of the international conference. But since the management of the Nehru Memorial College was supported in all the ways to conduct this conference, so we thought this is the time and conduct this conference in Nehru Memorial Colleges. And the remote place, the college is being run in a for the downtrodden peoples, I can say, for the education, giving the education to the people. So I am very much thankful on behalf of the organizing committee and Dr. Engineer Balasubramanian to support, because we need a financial support. That he told this much amount we will give. So that is the initiation. We started this conference and the support of this zoology department, particularly Dr. Saravanan. So we thought we contacted the resource person and sending the circular to the other colleges. So we are getting good response from the conferences, and especially Holy Cross College, Bishop College, EVR College, our own university, several universities in our own department. You see our department, life science students, all they were responding to attend this conference. So this is the good response we are getting it. So now the conference is being held. So I on behalf of the organizing committee, I extend a heartful thanks to the management of the Nehru Memorial College for giving the permission and conducting this international conference for the benefit of the students. I am also equally thankful to the administrative authorities of the Bharathasan University, Head of the Department of Animal Science, and the Vice Chancellor, Registrar. All they approved and given full permission to extend the support to conduct conference. So in that aspect, on behalf of the organizing committee, I extend a heartfelt thanks to the Nehru Memorial College. This is a joint conference to the Bharathasan University. That way we are very much happy about the conference as well as the response from the speakers, invited speakers and the students. Without the students, it is not being. So for the benefit of students, it is being organized. And later, we got a good response. We only sent our request to get the financial support to Indian Council of Medical Research in New Delhi. So a few days back, we got a good response. They sanctioned the partial amount. It is a very great. Please clap it for the support given by Indian Council of Medical Research. So every conference, we get support from the national level funding agency. It is ICMR sponsored international conference. So really we are very happy that the sponsors given by the Indian Council of, that is the engineer, means a president of the management is initiated the fund. Now it is again supported by Indian Council of. We are all very happy about the support or financial support received from Indian Council of Medical Research. That 2 days back only we received it. In that aspect we are very much happy. So Nehru Memorial College is one of the pioneering educational institutions. 50 years, golden jubilee, they have celebrated long back. They have started with means Puthanampatti. So Puthanampatti Ayya he started this institute. He means a leading educationist in this village. Now many people from village, if you go to any village, means I will join with they will tell about the Puthanampatti College, or they will prefer to Pundi College. So I studied in Pundi College. People coming from Trichy district, Kadalur, Natharkad, Salem, means they have studied here. I know it. And it is one of the, I am very proud to say that the computer science, the BSc computer science in the whole nation started this college, I know, next to the Bishop College. The computer science long back, we were not knowing how to start the computer science course. But very boldly it was started in these colleges. Now the computer science development, you know, everywhere in the globally it is very important. So that way the Nehru Memorial College is doing an excellent job, and several principals they have done in a very good way the administration. Now Dr. Pon Perisamy is a computer science. Again he is leading a very good, means running this administrative effort with the support of the management. So Bharathasan University is having a many several departments, and our own department is having a many roles, means diverse area of research. So this is the collaboration started with this department of zoology, Nehru Memorial College, and our university department. So it is a happy occasion. It's a joint program that is being held in the Nehru Memorial College. So we are all very happy. And the speakers, Dr. Padmanabhan, is very busy and one of the pioneering research in the cancer biology. Several years he has spent at Stanford University, and he is NTR Deputy Director, and he is a very helping man. Any students, Bharathasan University, at least 10 students they went to NTU and they have done for the project work. Any MSc student or MTech students. And Paul Murugan, he is also from our place, but he is in Stanford. And he has also agreed to give this talk. Hope he will give a talk. So like that, another Kanagesan from Malaysia, he has agreed. Then speaker, Dr. Ramalingam, who is sitting here, he is scientist in Tuberculosis Center, National Center, Cherpat, Chennai. And Dr. Debashish Das, chief scientist, Narayan Nethralaya, stem cell research in eye. He will be coming, he will be joining it shortly. Then Dr. Sanjeev Kumar, a biochemistry, leading scientist in biochemistry, and Rayalaseema University, Kurnool. Then 2 plenary lectures are there that will be given Murugeshwari and Lenin. So I am very much thankful to these speakers. So invited speakers, they are all readily agreed to give their talks. So I am very much, I also join, means welcoming the speakers. And the topics, biomedical research, fatal diseases nowadays, how to control it, diagnosis, treatment is there. So that way, the areas which cover here is pharmacology, biotechnology, nanotechnology, stem cell biology, biochemistry, bioinformatics, immunology, and chemical signals, microbiology, molecular therapeutics, and wildlife disease. So miscellaneous areas to be covered will provide us cumulative ideas on current techniques of biomedical research from animal to human level with ample illustration and results. So the idea is invited senior scientists are coming and delivering the talk, and junior scientists are coming and delivering the talk, and young scientists, research scholars and students are allowed to give you a oral presentation as well as the poster presentation. So time may be limited, but you will interact it. When the senior scientist is giving, there may not be time to interact with him, but leisure hours, when the lunch hours or tea time, you please interact with the people and get idea, get clear your doubts. So you have a small doubt will be there. So this is the time 2-day period, 2 days. So this is the idea to make students and senior scientists to discuss and to have a exchange the ideas, clearing the doubts. So that way it is arranged. Hence the total abstract so far we have received 94. So we were trying to reduce the oral presentation, but it is very difficult. Oral presentation we have included 26 and poster is 68. Today is only the poster presentation, 3:30 to 5. You just in the lunch hours you paste it and you go on. So oral presentation also total 94. So voluminous response, good response from the students. So it is the time to interact and all the things. So once again I am very thankful to the college management as well as our university authorities for permitting this, and you all just have interact and good things. You may have some difficulties and you bear with us, maybe with me or surroundings. So you just any difficulties you tell, enjoy, and it will be a very fruitful day for today's conference. Thank you.
On behalf of the organizing committee of this conference, we would like to thank our well-wishers. Thank you. Now this conference moves to presidential address. A man who is an inspired and leads our college strategically impact-driven with global exposure. All his endeavors and activities are marching towards the enhancement of this institution to high scale. He is none other than our principal. I invite our principal, Dr. A.R. Pon Periyasamy sir, to deliver the presidential address. Please, sir.
Good morning to everyone gathered here. Most respected President of our college, Engineer Pon Balasubramanian, respected Professor Dr. Padmanabhan, Deputy Director, Nangkang Technical University, Singapore, respected Professor Dr. Emmanuel Rajan, Professor and Head, Department of Animal Science, Bharadasa University, Tiruchirappalli, respected Professor Dr. Arjunan, UGC BSR Faculty Fellow, Department of Animal Science, Bharadasa University, Tiruchirappalli, Dr. Ramalingam, Senior Scientist, National Institute of Research, Chennai, Head Department of Zoology of our college, Controller of Examination, Dean of our college, Head of the Department of Botany, Dr. Sasi Kumar, Dr. Organizer, Dr. Saravanan, Dr. N. Ramesh from Aided, Ramesh from Self-Finance, other faculties, Dr. A. Venkatesan, Librarian, other faculties from Department of Zoology, participants from various colleges, universities, teachers from various universities, colleges, my dear students, media people, guests, good morning to once again good morning to everyone. Department of Zoology of Nehru Memorial College is the #1 department in research activities. First of all, I congratulate all the faculties in the Department of Zoology for doing better research. I am very happy and proud about the international conference on molecular advances in biomedical research. I appreciate the organizer, Dr. Saravanan and Dr. G. Arjunan, for arranging such a type of conference in our college. Nowadays, we are facing several medical related problems, disease in all age groups throughout the world. This has to be solved by advanced scientific methods with the minimum cost. A common forum is required to discuss about the recent trends in medical sciences. Hence, the participants can update themselves by attending such a type of conferences. I congratulate the management, I congratulate the organizer, I congratulate the participants, students, Department of Zoology. Once again, wish you all success for this conference. Thank you, one and all.
We have Dr. P. Puthanampatti, sir, Deputy Director, Lee Kong Chiang School of Medicine, Nanyang Technological University, Singapore, as a special invitee to this inaugural function. He is well-known academician and a researcher, and he has chaired many international and national conferences and seminars. He contributed his ideas and knowledge to the scientific world by way of publishing and presenting articles. We are very proud of having Padmanabhan sir on this occasion. May I request Dr. P. Padmanabhan sir to deliver inaugural address? Please, sir.
Good morning. I was bad throat today, I don't know. I have to survive. So, respected visionary, because I want to start with the visionary, Professor Ponnambalam, Chairman, Governing Body. Respected Engineer Balasubramanian, President. So respected my beloved brother and friend, Dr. Arjunan, who is, he is my senior, I have to mention here, 3 years senior than me. And he inspired me to do research. And Principal Dr. A.R. Pon Periyasamy. So, Sri Pon Ravichandran, Emmanuel Rajan, and Kanagation, my friend Kanagation, Professor Paul Murugan, Dr. Ramalingam, I think so one and all in the list. So this international conference basically to enrich the knowledge of the students in particular. The reason being is very simple, because we are in a rural environment and we cannot take it all the idea of what we are globally having as a pool. So this kind of conference, the scientists or eminent researchers, they feel that by the way we can reach even to the nook and corner of the world. This is the concept behind why the international conference has been organized throughout, and every time you see nowadays the Google, you can Google any topic and you can get the numerous conferences running every now and then and every day even. So here I start with a simple thing. Because being a student, being a rural, I born in a small village called Simutlu nearby Chidambara, because normally I tell my story also to inspire the people, young students. Because those days, if you join zoology, the people will think, everybody criticizes. So zoology is the last option. Because if you don't have option, then the last course is the zoology. It is not the case actually. Because the life science, it starts from the life. Because everything from the bacteria, we well aware, from the virus, because nucleic acid. So it is very important to keep it in mind. It is not in the course. It is only how you are looking at. Because your vision will promote and you bring it up to the level you want. If you work hard, then definitely that will brings you the fruit. So with that thing, I want to start with the thing. Just I want to begin with a small, because in interest of the time, I don't want to go in a beyond way. I thought that I will talk more than 20 minutes, but I will cut short to 10 to 15 minutes. The Human Genome Project is the boon for us. So why? Because we know that there are 20,000 annotated genes which are responsible for producing diseases. So I think this is apt to mention here. The reason is very simple. This is a biomedical science conference. See, this out of 20,000 annotated genes, whether we have medicine for all 20,000 annotated genes? No, we have only 10%. So this is our bound duty to search for the rest of the 90%. That means, so another 16,000 annotated genes are left over to find identify the disease causing mechanism and to fix them up. Then our human health, we can very proud and say that our human health will be get improved in a way. And in that, there are several contributions. So everybody now talking about, like a doctor, if you go to the doctor, they say, I am specialist only for the left eye. So I am specialist for the right eye, like that. So that means we are growing in science in a focused way. So what it tells us? It tells us we are focusing in one area. We want to focus on that area particularly to get success. That means if a proteomics person, they talk about the proteomics, then his duty is to identify the biomarker. That's what we are talking about. Everywhere you see, there is a biomarker approach. So why is a biomarker? So earlier we talked about the antigen, antibody, epitope, all these things. Now the biomarker approach is very useful to identify and target the particular disease. That way you can do the diagnosis as well as the therapeutics. So that it is called the theranostics. The new terminology has been coined. And you're going to see the new terminology going to come in few days, which is called the mechanogenomics. That means if any pressure applies, then how the genome, it will be altered. So it is a very latest field that you will see in the paper soon. So this is the transcriptomics. So how the gene will be transcribed, we are talking. And we are now going in detail how the transcriptomes contribute to the disease and how it is changed during the course of time. So the person is normal up to 40. Why the person is diseased at the age of 45? We don't know the answer. So that means, so we are, going into the deeper, deeper and deeper, and we are searching the cause, how it is happening. So then the next level is, we are looking at the interdisciplinary. Because now, golden days, see, I am a zoologist, don't come to my area. I am a botanist, don't. So this is my plate, and this is my food. So not now. Because if you are interdisciplinary, then only you will be successful. We need engineers to design a machine. So in the later stage, I am going to, I restrict my talk not touching the imaging, because I am going to show some of the things. So how many engineers involved, how many engineers means on the whole, electrical, electronics, so all engineering, including civil, because if you want to have a radioactive shielding, you need to have the lead bricks, and you have to have some cement, concrete shieldings, so many things. So all the engineers are in place. So that is why I want to say this is a multidisciplinary. If you are multidisciplinary, and you are very successful. For example, I am a zoologist, but even I will talk a little bit about physics, engineering, all that. So because it is the interdisciplinary approach is coming, and that way, even in any school, doesn't matter, even small college or hospital or big university like NTU, but you have to come closer and you have to identify the area of the research to work together. That way, success is on your way, as well as you will do better things, new things. Because you are talking about only the same thing. It is known thing. Because we are repeating the same experiment, like protein analysis. Yeah, everybody does total protein, Lowry et al. So this is all, the old story. So we have to come closer, every department, in an interdisciplinary way, and do the research. That way we will move forward. In interest of the time, I stop my talk here. I want to say a message to the younger generation that all you just to be confident and do your work, sincerely, and you will be successful. Thank you very much.
Our president works with high thinking of developing and changing the students' community by implementing innovative programs which boost up their same. He works 24/7 for the welfare of students and the college. All his efforts are around the development of society, in particular the country in general. May I request our President A.R. Balasubramaniam sir to felicitate the conference.
Dignitaries on and off the dais, faculty members, research scholars, and my dear students. I don't want to take much of the time because we are hard pressed, and 2 days I don't think it's sufficient to conduct such an eminent research-oriented international conference. So, at the outset, I congratulate the Department of Zoology for organizing an international conference. I'm sure the deliberations will go a long way in shaping the future generation of researchers who are all sitting here in front of me. The idea is, you cannot learn everything within this 2 days conference, but you have to create the network. You have to meet the people, resource persons like Padmanabhan or Dr. Ramalingam and other research people who are coming from various institutions, and you should see their line of thinking or where the research is heading to. The trend is now, as the theme rightly points out, molecular advances. We are now going deeper and deeper to the molecule level of rectifying the diseases, be it cancer or other diseases. So it all has to help the human society in large. So all your research, I want you to focus to make a social change, to help the people live better and longer. I once again congratulate the department and also thank Bharadasan University, Professor Emmanuel Rajan, and our own Dr. Arjunan, because we don't feel him as an outsider, we always feel him as our own college professor. Thank you, sir, for helping us to organize this seminar here. I'm sure it will go a long way in helping everyone. Researchers, one suggestion to you is, you have one thing higher than what all these resource persons have here. You have a lot of time. You have to identify the areas with the help of the resource persons and put your concentration and do your research in that area. I'm sure you'll be successful in the years to come. God bless, all the very best. Thank you.
The Department of Animal Science, Bharadasan University helped us a lot to collaborate with the department. With his support, we organized this conference. I request Dr. Emmanuel sir to deliver felicitation address.
Very good morning to everyone gathered here. So as a participating department of this international conference, I am here to facilitate the initiation of this international conference. So first of all, Dr. Saravanan and my own senior Dr. J. Arjunan, so I really congratulate them for making this great effort into a big success. And then the administration of this college, they are the one to working 24 hours and all the days for the welfare of the students. So I congratulate you, sir, for making this positive thought for your the new growing lives. And then the principal and for making this big effect, and all the invitees, for example, starting from Dr. P. Padmanabhan to the president, and the another one here is like Dr. Ramalingam. Everyone are really traveling far from to this place. You can imagine like how much effort they are taking and how many times they are spending to reach this place. It just only because of the participants, those who are sitting here. Okay. So I wish mainly all the participating students, hope this 2-day conference will enlighten you and will take you to the different heights in your scientific endeavor. Hope this will be the first firing spot in your brain that takes you to the different directions. So I wish you all the best. Please make use of this, and thank you.
Beloved Chairman, respected President, Honorable Secretary of our college, respected Principal, our most valued invited guest, and resource persons of today's function, teaching and non-teaching staff members, and my dear students. Good morning to one and all. It's my bound duty to express my sincere thanks to our President of our college. He is the motivator and pioneer to all the staff and students. Next, it's my duty to express my sincere thanks to the Secretary of our college for motivating us to arrange this event. We are grateful to the Principal of our college for accepting our invite and providing necessary facilities. On behalf of our college, may I take this opportunity to thank the chief guest today function, Dr. Puthanampatti, my dear friend, Deputy Director, Lee Conley, School of Medicine, Singapore, for having come and delivered a fabulous and valuable inaugural address to the gathering. On behalf of our department, I feel happy to thank Dr. G. Arjunan, UGC TSR faculty, Department of Animal Science, Bharatdas University, Trichy, for joining hands with us to organize this international seminar. A big thank you to Dr. Emmanuel Rajan, Head of the Department of Animal Science, Bharatdas University, Trichy, for taking more efforts towards this international event. And also, it is my duty, express my sincere thanks Dr. Priscilla from Bishop College, Dr. Ramlingam, scientist from NERT ICMR, and Uma Maheshwari from EVM College, and Pushpa from Kaveri College. They came here, they are our friends. Thank you very much for their arrival. Next, I am very happy to thank our beloved students and participants to be here and make this function will be the successful one. And one more thing, Dr. P. Nehru Narayanan, now he is the controller of our college, but he is controller of our college, but also not only the controller, but also our department eminent professor. So I must thank Dr. Nehru Narayanan sir. Before I conclude my valued vote of thanks, once again I thank you to one and all for presenting and showing for encourages in this event. I also acknowledge the contributors, all those who are going to present papers at this international seminar. Finally, I would like to take this opportunity to place on record my hearty thanks one and all gathered here. Thank you very much.
So now we are moving on to the important session for this conference. So for this first meeting, I'm happy to share this session, and thanks for the organizing committee to do this job. So now let me introduce the speaker, Dr. Puthanampatti, and I'm privileged to introduce him. And he is an eminent scientist who has an experience of more than 25 years in science that too in a reputed discipline and in the various in the institutions internationally and in a reputed design. So he currently he is a senior scientist in Singapore Nanyang Technical University, and earlier he has been working in Stanford Medical School. It's a prestigious institute in the USA, and he has been held as a different positions, like as an acting position, professor, visiting professor in Bharathasan University also. And his qualifications are like he is a PhD from Annamalai University, and he do also have a different editorial role in various disciplines, and he is a reviewer for several international journals, and his current research interests are molecular imaging and discovery. So besides, like, he is a member in a lot of professional bodies like the ASM, American Society of Microbiology, and a lot of microbiology societies and Life Society of Bioethics and other regulatory authorities. And he has more than 100 publications. And he is active participants in the academia, and also like he is a member of academic and industrial expertise. So now I call upon Dr. Puthanampatti to come and deliver his talk. Please welcome, sir.
Thank you, sir.
Thank you very much for the nice introduction. And I came to know that where my cousin brother is working in the same organization, he is also working. He worked, my cousin brother was there. And I have to say here, because the young audience, you are, I don't know how many people are really Nobel laureate sitting here. And this may be a little bit inspiration to you all. Can you project? Give your insight on translational molecular imaging in biomedical research. So there are every country now, making a biomedical act, which is important to the country growth. So here I am going to, emphasize or give an idea. So what is really going on in the globe? So what is I'm going to cover this area? What is molecular imaging? So what is the translational molecular imaging or translational molecular imaging? And technology platform, what you need to do such kind of research and application of molecular imaging. So here, molecular imaging, it is nothing but molecular processes in bio, and it is in vitro and clinic and finally to the bedside patient. So all the clinics means it is in the hospital setting. So the molecular, you know that everybody, the cell is the basic fundamental unit of any living organism. So the cell has several organelles. If you see that organelle role, for example, endoplasmic reticulum, if it is rough or smooth, the rough endoplasmic reticulum involves ribosomes, which is a protein machinery. So like that, Golgi complex, which is secretory in nature. So what really we are looking at here, in a collective way, if it is a secretory cell, so many Golgi complex will be there. That way, the secretory things will happen inside the organ, inside the cell that deliver, and you can see in a collective manner. So in vitro is one of the the technique. So you can culture outside the body of, especially the cell culture technologies available now. Even animal cell culture, we call it as collective tissue culture. Then the same way you can collectively get into the clinics. For example, if you are getting some result in vitro culture, for example, 5-fold increase, then you can same thing you can apply and you will get 1-fold increase in the living system because of so many complications like excretory system and other things. The background I already mentioned in inauguration. The Human Genome Project had a tremendous impact on the medical practice, especially the biological research has, the 21st century was taken further because of the, Human Genome Project. Some of the diseases like cancer, even the tuberculosis, because now we are doing PET imaging on tuberculosis because it causes only by the, happens by the bacteria. So then the gene therapy is another, the thing how we can be able to control the disease. And for example, I want to give some history of imaging. The Roentgen discovers X-ray in 1895, but it takes about 100 years to put the PET and CT together. That means then the computer tomography has been invented, and how the snapshot, the X-ray technology has been computed. It's a tomography patient. Then finally, in the year of the 2000, David Thompson, who is my mentor, he put positron emission tomography and the computer tomography together. That way, in every hospital we are seeing now the PET-CT, which is one of the important modality to diagnose cancer and even for the angiography, all other technologies. So multi-modality imaging. What is a multi-modality imaging? So if you are doing one image at a time, that is called one modality. For example, optical way of imaging you are doing, that means optical imaging. If you are doing a MR imaging, this is another modality. If you combine these two modalities, you will get a better understanding of the disease problem. So when we are advanced in biology, the medicine needs, especially the diagnostic tool, very precise. Otherwise, we treat the patient in a wrong way. So for that reason, the multimodal imaging is very important. So here are the types of imaging and what it really gives us. That is, can you switch off the light? So the CT is a computer tomography. The computer tomography gives anatomic information. So that's why if any structural image you want, the CT will be done. Then they will look at any changes in the structure. Then some of them, we are doing ultrasound. For example, the ultrasound also gives the structure, but is in a vague way. But it will give little bit molecular because of all the motions you can catch in that. So the MRI is another technique. By the way, you can get anatomy, physiology, metabolism, but molecular level is very less. But in the way of nuclear medicine, especially, you do positron emission tomography or single photon emission computer tomography, you can able to visualize all the anatomy, except anatomy, you can see the physiology, metabolism and molecular level. The thing is very simple. If you combine two modalities, that's why the PET-CT is very successful in a hospital in diagnosing, because two modality gives entire picture of molecular structural information. The optical imaging is very good, but it is only suited for the preclinical, not for the clinical. The reason is very simple. We cannot apply any fluorophore inside the body, which will be the toxic to the human being. So that's why optical imaging is not really in practice in clinic, but now they are just finding the way how they can improve and use in the clinic. So here the potential roles of molecular imaging. So we look at all the techniques. The technique itself is not sufficient to do anything. So you need to understand first, so what really it is suited for. I already mentioned that the optical imaging is best suited for the small animal imaging studies. The micropet is the PET version of the animal, which is called micropet. If you do the human PET, that is called PET. So micropet is the, basically, because I am talking about the translational, here the micropet will give the anatomical and, the especially functional details of any living organism. So these are all the things, potential roles of molecular imaging. Just I am leaving in the interest of time because the 45 minutes was given to me. Then what is the translational molecular imaging or molecular neuroimaging? Then here the translational research needs, the researcher should design and concentrate on bench to bedside research. So what is bench to bedside research? That's why all the university, they are getting fund, and that from the fund they concentrate only on the basic research. For example, they do on some molecular level, on bench studies. For example, the DNA may be taken, they do different modifications or manipulations, and finally they put it into the bacteria, and the same gene construct will be put into the animal for the gene therapy purpose and other reasons, and finally you will get something. And the genuine reason, technology and applications will promote, collaborate with the other groups, can contribute to translational research. So that means it is not only, with the technology application, but also it promotes the collaborative nature of the research. I already mentioned that even interdisciplinary, the research will be more fruitful than other research. And efficiency reasons, direct transfer to the basic knowledge into medical applications. So if the research question is correct, then you can take it to the bedside, That way, what you are doing, which will be, preclinical way of assessing the disease, and after that it will be tested in the human. That is why we have so much clinical trials. The NIH, National Institute of Health, is more focusing on clinical trials, and if any molecule or synthesized for the, synthesized as a drug will be passed into several categories, and finally it will be given to the human being. So that's why the trials are. And economic reasons, of course, you are seeing all multinational companies, multinational pharma companies like Novartis and all, they are pouring money like anything to get one single molecule. The reason is very simple. When they are successful, it will be completely translated to human, and they will make money like billions. So this is the reason. Of course, everything has economic reasons, but the basic consideration is how we can improve the human health. And the translational medicine, it is efforts to carry, again, I want to put as a bench to bedside. It builds a basic research studies of biological processes using, example, humanized animal disease model. So why humanized? In olden days, we experimented only with animals what we have. So our, the wild animals, like a wild rat, wild mouse, like that. So now we have an option of having a transgenic animals. We are talking about transgenic animals. That means humanized transgenic animals. So what is the gene? Which gene is responsible for producing some disease? That particular gene itself will be taken and given to the animals. You have to clone at the embryo stage. That is called transgenic animal. What it does is that transgene will express and mimic the same disease mechanism in the animal. So these animals are transgenic animals. These animals will be used, and they will give all the trials. So whatever you want to test in the human, the same thing can be applied to the animals, and finally you will conclude whether the humanized animal model is behaving right for the drug or for the molecule. So that way it involves the transformation of bedside, finding into new ways of diagnose and treat patients. That's the key actually. So you have to diagnose, and if the disease is correct, then your treatment, what option you are selecting also correct, then finally the patient will go home with the cured condition. It helps predicting, preventing, diagnosing and treating the disease. That's what I explained in a bigger way. It aims at accelerating the bench-to-bed process regarding the application of novel knowledge about the physiological, pathophysiological, all things. You can notice, what physiology when you are giving a drug. I don't want to touch the new area like artificial intelligence. Even your heart will be taken and put it into the computer, and you get all the information, and you can even do the dosage in the computer and control your heart after that. So this is called artificial intelligence, which is coming. We are collaborating with Princeton University for that. So this kind of research are coming up, and in future we may have a better the medical toolbox for each and individual person to cure their disease. And basic and clinical research are interdependent, because you see the basic research, without basic research we cannot move on to the clinical research, and when the clinical research is successful, then the improved health will be seen. But you see that the translation from basic science to human studies and translation of knowledge into the clinical practice. Because that is why, other country, each and every doctor should, they do research. It is compulsory. Or, they will select other way. The doctors will prefer the clinic only 10% and 90% they do only the research. The reason is, if you do research, you understand more about the disease and you can treat the patient in an efficient way. And I want to show some of the, the thing, NIH budget of 2008, how the money has been spent by the NIH on the clinical research. And the non-clinical research, that means the basic research, is around 68%, 67 to 68%, and other clinical research is only 20%. You see the clinical trial, only 12%. That means that they are giving more importance to basic research. By the way, they can get into the final product, very, narrowed down, and it is very less, 12%. Even at the end, it may be 1%. After all the clinical trial has been done. And the need for translational research, because the molecular imaging biomarkers, now we are in the era of omics era. We are having a proteomics, genomic, proteomic, metabolomic, and even finally the clinical applications. And the disease modifiers and drug. For example, if you are treating any disease, you have to understand one mechanism of the particular disease. How you understand? You need to have disease modifiers. So, for example, you have to create a disease as well as you have to control it. How you can control that can be done only with the preclinical animal models. So, you have to do first the creation, and you have to somehow manipulate and manage, and finally you will be. That is why you have to add the blockers. For example, you are working on one receptor. That particular receptor behaves in a way that means you will want to treat whether receptor mechanism, it is not non-functional or it is malfunctioning. So that point of time, you need to treat with the normal subject with their modifiers. That means it blocks the entire processes. Then finally you treat. Then this is what by molecular imaging we can see that. What you are seeing here, so this is the dopamine transporter. And dopamine transporter, when you block them, you cannot see any transporter in the striatum region of the brain. So that way, you can able to image the patient and you can do. And the translational medicine platforms. So far, so many platforms are available. Starting from genomics, now genomic is coming down and molecular imaging is going up. The reason is very simple, because NIH don't want any genomic data anymore. They want the PET data, positron emission tomography data. The reason is the positron emission tomography, the sensitivity is very high. Even there are false positive when you amplify, when do PCR, SAR may know. There are some false positive chances are there. So in order to, this kind of understand the mechanism behind this, they want that molecular imaging data, which is, GLP or whatever labs. And the animal models in translational medicine, there are many models. Again, the zoology play a major role. You see the vertebrates, you see the primates, dog, mice, rat, and zebrafish. The primates means there are New World monkeys and Old World monkeys, macaque, and even marmosets. Marmosets are called the New World monkeys, which has the similarity as like that of the human being, because the marmosets lives only with the siblings. So we are establishing the colony and breeding right now, and the marmoset research will be the big research soon in the whole flow, because this primate, non-human primate, will give more precise the values which will help the human disease control. And the invertebrates, the worms, and the single cell organisms, phage, bacteria, yeast, still we are using. So these are all the animal models we are using in the translational research now. Sorry. So the drug development, I told something about bubbling, then I want to show you as a cartoon how the drug delivery has happened. So for example, the synthesis and purification. So every pharma company, they comes with a million molecules, million compounds every year. And finally, they will give to the CRO to evaluate. CRO is the Contract Research Organization. So normally they do the high throughput screening. By the way of high throughput screening, they will identify the key molecules. Then the key molecules will be given to the handed over to the pharma company again. Then they will do many, like the steps, like all the screenings, the preclinical thing. Then it will go to the phase 1 trial, which will take 1 year. This is called the dosing with the healthy volunteers. Normally they choose 20 to 80 subjects. And the second phase will be 2 years, maybe 100 to 300 patients. And the third phase and finally auditing will be done. And all the 4 trial phases, then only the drug will be delivered in the market and it will be given to the human being. So the entire process will take around 12 to 15 years. And here one of the classical thing, because we are in the neuroscience and the neuro, especially translational neuroscience, so we are mostly working on from the rodents to the human being, and we have all the scanner for this, because this scanner is very sensitive, and even you can do PET imaging at the level of 0.7 millimeter resolution. Imagine, because the rat and mice, the brain is little, literally very smaller than the nail of our last finger. So that brain can be imaged, and what you are seeing is the dopamine transporter and receptor. So which is very, it only target to the point, and it gives a signal only from there. And the rest of the things can be get cleared, and finally it will be, comes out of urine. So this is the beauty of the molecular imaging, whatever you want to see. That place, that particular receptor means it binds only with the receptor and stays back there up to the half-life period. The disadvantage, of course, because the cost is very, not easy, because India, like country, still we need to have another 10 years. Even though in the big hospitals like Apollo and all, they are setting out the medical cyclotron, and they are producing every hour so many, like the nuclear traces, the radio traces. The traces, now we are only using FTG, fluorinated deoxyglucose, which is easy to monitor and get the images from the cancer and other abnormal tissues. The scientific basis of molecular imaging. So I already mentioned in my talk, there are 20,000 annotated genes are there. So the 20,000 annotated genes, only we have drugs for only 10% of the genes, and rest of the 90% we are still working on, because the Human Genome Project 2000 give us a tremendous idea how to improve and identify and treat these things. That's why even now the people are concentrating on mechanogenomics, and how even the pressure it modifies the gene content. By the way, it will reflect on the entire the processes. So here you see that there are drug modifiers, and like I already mentioned that. So you have to go the full 100% the screen. So you should not leave even one small bit. If you lose one small bit, you will lose some information, which will be the key. The control experiment is the key for developing a drug. So this is the scientific basis of the molecular imaging. So especially this is what the Human Genome Project, and we got the genes, and even we transcriptomic, all this kind of. Even now we went to the level of single cell the sequencing. So by the way, you can able to, even one neuron is malfunctioning, then from the neuron you can able to find out what is the cause that neuron is not functioning. That way of genetic information you will get now. And what is the translational molecular neuroimaging? Especially, see, when you are going to that, as the radio-labeled probes, which is more precise, and some of them we will start the reverse translational process. What is the reverse translation? So they start from the human instead of from the animal. So it is very simple, because if the person is diseased, so the brain will be harvested in our case, because we are related to neurobiology. So the human brain, So immediately we have a program called Brain Bank program. So what will Singapore, if anybody is registered, that they are having Alzheimer's disease, then the brain will come to us. Then immediately the body will come. Then we harvest the brain and keep it in -70. And whenever we want to, have some, this kind of study, we cut the piece of the brain and our developed new probe will be applied. It is called autoradiography technique. It is a very old technique, but still it will be very helpful. Then, if it is binding with this kind of the Alzheimer brain, the tissue, then what we do is we will reverse it, go for the animal, the same probe, and we will come up to the brain. So this is the way how the radiolabeled probe, the molecular imaging and molecular probes will be developed. And that is what I mentioned earlier. There is a bench to bedside. There are two ways of the thing. One is the back translation and forward translation. So when you start from the tissue culture and taking further to non-human primate and clinical trials, phase 1 to phase 4, which is called forward translation. When you are doing like what I shown before, autoradiography, then you go back and inject from the animal, and that is called backward translation. That way it will, some people they want to, some lucky people they will get, quick results. And the possible contribution of various translational medicine research platforms. Here I took my own school as an example. To look at it, all the schools here, the School of Chemical, Biological Engineering, Electrical and Electronic Engineering. See, all these, the schools will contribute to the entire molecular imaging. So that means the translational molecular imaging demands the multidisciplinary approach. That way, the success is 100% and you will do good research. And the technology platform, of course, you will see. So far, I have just told the story in a theoretical way. Now I want to impress you whether my statement is right or wrong. Do you have any evidence for all the things what I have told you theoretically? So here you see that there are high sensitivity PET scanner, HRT scanner. The community, entire research community decided make only 10 scanners, distribute throughout the globe, and see the results. Same the kind of task was given, and they just scanner performance as well as how the probe is behaved. So this is where the way the scanning of human brain with high resolution, the scanner was developed and it was given to the different top-notch institutes, and the resolution of sensitivity is around 1.5 millimeter. Because in human being we can get, because 1400 gram, in mice it is 1 gram. So 1 gram versus 1400 gram. So you need a scanner 1.5 millimeter resolution. But still in animal scanner we can achieve only 0.7 millimeter, which is not in the scale order. But anyhow, the 0.7 millimeter even we are very happy to note. And this is again the platform development in the current status. You can see the scanner like this. If you want to do mouse or rat, this is the scanner which cost about our cost around 5 crores. And this is a scanner for monkey which cost about 7 crores. And this scanner will cost the same. Even the small scanner also same price, but even human scanner also costs the same. The reason is because the technology is the same, but you have to other things, especially the components, the machineries, or especially the circuits and other things, or the things, crystals they use, all are in the high order. In the arrays are different. We need at least 3 rings of the PET rings we need in the machine of mice and rats. And what you are seeing here, the same the thing like the striatum. This is called the dopamine transporter area, and this is the rat where you are seeing. This is the non-human primate, that means monkey. This is the human. You see, that is why the similarity between the monkey and the human is the same. That's why the non-human primate research is considered as it's closer to us. That way, any drug work with the non-human primate will work with the human. That is analogy, but we also fails in some of the things. And this is the scanner for mouse and rat, what we have. And this is from the Mediso, the Hungarian company. It manufactured. This is the topmost one now in the current market. And this is the interchangeable the animal beds. So the same animal can be shifted to the thing. You can do PET CT and also you do PET MR at a time. So that means you will get more the anatomical and functional images. Detailed one. This is the cross structure. So this is the one, if you want to do multimodal imaging, so you can do the one step. You can do all modalities. See, these two scanners are there available with you. You can have this one. You can get a structural thing, bone image, and you will get only the tissue image, functional tissue image, and or the bone image. And this is by MR. You can get the soft tissue image, and you can even superimpose all the images, and you will get the whole information. That's what I have shown the cartoon, saying that what it is good for the one modality. So now I combine all the modality, and what you are seeing is the collective information, and you can able to get. And here is the sensitivities and resolutions, because normally we have to look at it. Every paper, when the development comes, any technology, the technology demands the precision. So how precise it is for your research, so that it is very important to understand the spatial and temporal resolution of the imaging tool developed. So this is the imaging performance, the nanoscan PET CT again, only the bone scan. This is the fluorine 18, which is the half-life is 110 minutes. So I want to stop and tell one small note here. So we have a positron, we have electron, right? So we also have the positron. So the positron and electron, what is the difference? The electron is negatively charged and positron is positively charged. So when we put the molecule, especially F18, water, in the cyclotron, when we bombard, we will get the positively charged molecules, which is radioactive in nature. The positively charged molecule will be injected into the body. Our body contains so many electrons, it binds with the electron and annihilates, and it gives 511 keV energy in opposite direction. So one photon on the other side, other photon on the opposite side. So these two photons will be collected in the scanner. That is what you are seeing as an image. So this is a very, but one interesting thing is, these positron emitters half-life is very short. So the patient will go in the morning, injected, and the evening they will go back home. That means 5, 10 half-life. 10 half-life will be done. Then there is no radioactivity in the body, which is safer. So this is what, this is a bone scan. And also if you want to image the heart, and you can do, like the positron emission tomography as well as the MR image, then you superimpose, then you will get this image. So what you are seeing is the real-time image. The real-time means when we inject the tracer, in tail vein, in the case of mouse, this is the mouse heart, injected with the FDG, fluorinated deoxyglucose. So what this FDG is good for? The FDG is good for the glucose metabolism. So what you are seeing here, it is not the movement of the heart. I would say that this is how the glucose enter into the the heart is functioning, because it needs a continuous supply of glucose. By the way, only it will get energy to contract and relax. So what you are seeing, it is an influx of the glucose getting into the tissue for the function of the heart. So this is the beauty of this molecular imaging. Whatever I explained, this is the one example you can do for the cardiac imaging. So we are in the neurobiology, so we want to have every receptor in the brain, want to image. So this is the image of the mouse. And like the dopamine transporter, dopamine receptor 2, and the striatal imaging, FDG, fluorinated deoxyglucose, FEP2I, that also for the transporter, and even the C11, FLP, all will be focusing on the receptors of the brain, especially dopamine receptor. And you are seeing on the right hand side, this is the real time again. how when you are injecting the tracer in the body and how it gets into the brain and how it gets cleared. After that, you see the clear image that is taken. This is the 0 minutes. So what the animal will be kept in the scanner, we say that dynamic image. Push the button, then it continuously collects the frames of the image, and because of the computer application, we actually process the image, and finally we will get the images like this. And the application of the molecular imaging, there may be a question, so you can image a receptor, you are telling. So do you have any evidence whether you can able to guess? The estrogen receptor, it is mainly, studied. It's a well-studied receptor. It was crystallized in 1997, which is reported. And this all normally forms a homodimer. So it is in the cytosol, but this is called a nuclear protein. So we call it as the nuclear protein because it produced, translation and translate. When they translate in the nucleus, then it will produce. So, for example, this will show. This is the estradiol, which is important to form a homodimer. So here there are two estrogen receptors. How the homodimer forms? If the estradiol didn't bind to the estrogen receptor, there is no homodimer. That means if the homodimer should happen, either it should have the agonist or antagonist. What is agonist? What is antagonist? Agonist is the one which triggers. Antagonist is the one which suppresses. So what here, even though estradiol is there in the free form, it should attach with the receptor, then only it will form the homodimer. So now it binds with the receptor, then it forms a homodimer. Then it goes to the ERE in the nucleus, especially nuclear ERE means estrogen responsive elements. It sits there, then it activates the transcription. So then it transcription starts, then it will translate into protein, and finally it produces. So we just made a split reporter technology. The reporter technology means the reporter reports the cellular event in vivo by the way of some means we can able to visualize. So for that reason we taken the renilla luciferase. Luciferase is from the firefly. This is from the worm, renilla luciferase, and we have to inject the substrate. So then we made some gene construct. So how? It also like this, and it binds with the reporter gene, then it binds and it produces the light, but it is not working. So these are all the the different constructs made, gene constructs, and we tested with the control. So the agonist as well as the antagonist. So what you are saying here is the estradiol is agonist and the tamoxifen is antagonist. The tamoxifen is the treatment option for the breast cancer. They give this. And you can see finally, because the tamoxifen and the relaxafen, which are the drug which is treated for the breast cancer, which gives the suppression, and the estradiol which gives the activation. So this is the difference between the antagonist and agonist. And we done all the things, and finally we did that. So then the other question will come, whether we can do the stem cell research, and by the way we can use the molecular imaging technology. Yes, we can use. So normally if you do any bone surgery, especially in the fracture happens, there may be a 10% of malunion. The malunion in the sense that the patient will be, everything is perfect, but there is not a real walk like before, or real, the movement like before. This is because when they are placing, we are the human error normally happens, in a millimeter order or something. So it will really, matters. So when you treat with the stem cells, it can be avoided, because the stem cell, it's every stem cell, has the, tendency to, develop into a lineage, like, even the adult stem cells, the fat cells also use bone, cartilage, neuron, and muscles. So we took this concept and we made a gene construct, and we ordered a triple, the construct, the tri-modality construct. This is where in order to monitor in different modalities. And finally, we just injected to the mice. We made a defect on the bone in the real model with the driller. the driller is very small, and we conducted in Stanford because, very precisely. And then we did the images, injected the stem cell to the treatment affected animal, and finally the image. And we could see this is the optical imaging. It looks like a noisy. Then finally to analyze every animal, and you see on the day 7, you can see the more recruitment of stem cells to the affected area. So then, then we did the CT, and by CT also we see at the day 7, you see the control versus the day 7, the treated animal. You could see the bone formation clearly here, and finally we did the even PET image. Then you see the gaps between the day and the day 10, because day 7 our some cyclotron problem, then we switch over to day 10. So these are all the things, finally, in order to confirm, the golden standard methods. So you can even section the bone, and you can able to see adding the substrate, because here we use the firefly luciferase. Firefly luciferase kinetics is very long. It is 28 minutes long kinetics. So we used that one, and finally we could see there is a treatment effect. There is a stem cell. That's why when you add the substrate in the light, you can see the light, and whereas in the normal we could not be able to see. So these are all the different way of confirmation how you can do. The same way you can do the cancer, and how you treat the cancer with the drug, and how you get rid of it. So this is also the other study. And he's a nuclear imaging again. I want to say that SPECT. I don't know how many of them you know about the SPECT, single photon emission computer tomography. So normally this kind of technique in nuclear medicine is very good, and still it is valued like anything. Even for me, it is more fascinated. The SPECT is more fascinated to me rather than the PET. The reason is the energy level is low. So when the gamma becomes beta, you can have the theranostics approach. You can treat the same cancer. No need to apply again, and it will be when it decay to beta, it will give because of the long half-life, it will be act as a drug. So that way, this is one of the way. Even in small animal imaging, we are using SPECT in a big way, and I will show you some of the things how the GI tract can be imaged. For example, in the GI tract, which is very one of the considered as important thing, even though we neglect, we eat a lot of stuff, but our stomach and the intestine is taking care of everything. But at one point there is a lesion, and it complicates and finally leads to the cancer. Yes. So in order to see the lesion, we joined hands with the pharma company, Abbott, and we conducted some studies on the GI tract, and finally we this is how it looks like, the system of the mouse, and these are all the snapshots how it looks like when you collect the slit imaging, and that is the way you can be able to do. The idea here is I put the snapshots to understand the molecular imaging will help not only to the receptor imaging, not only to the the stem cell research, but also to the gastrointestinal things. And here, this is also a SPECT image of maximum intensity projected. And what here you are seeing is the sodium iodide symptom, because this is the image with the pertechnetate, which is called the technetium 99M. There are two names synonymous. So this technetium 99M can be used to image the thyroid. So whenever the thyroid cancer, there is ablation is done. So then this is the only technetium will be injected to the patient. Then it will be imaged. The half-life of the technetium is 6 hours. So this is the way it will be. So then this is the HMPAO. This is again the SPECT molecule, which you can able to target only brain, and you can able to see the lesion and other things. Okay, thanks. I'm almost. So yeah, so here I don't know how many of them. This is I put in order to understand, because most of them are students. So the sodium iodide symptoter. So the sodium iodide symptoter symport sodium inside with yes. Sodium iodide symptoter. 2-3 sodium will come out before entering allow one iodine inside. That is why it is called sodium symptoter. So I will repeat the cartoon again. So see, so 3 sodium will go and the 2 potassium came in, and 1 iodine will be guarded by 2 sodium. Again, this is the way this it imports the iodine. So when you do this kind of imaging in molecular imaging, actually it is possible. See, this is the beauty of the image. So when you do the positron image injected, and you can see it is co-registered with the CT, and you could see only the light up region is the thyroid. So thyroid is more populated with the sodium iodide. So this is a multimodal MR, so you can do MR. There are many kind of technology, the sequences available. By the sequence you can see even angiography, even grey matter, white matter, all these things. And in this nanotechnology also you can do. You can apply and do some, the imaging using this. Interest of time, I'm just skipping. And even you do ocular things, how the eye, if the drug is applied and how different it behaves differently, that you can do. And we have now, I am running the head of the operation of the Center for Neuroimaging Research at NTU. It was a big initiative, and we have 3 phases. The phase 1 has been developed, and we are having 3 Tesla MR, 3 Tesla Mach MR, and we have MAG, we have NIRS, we have even OCT, all kind of 8 different modalities for brain imaging or brain monitoring devices. And so with this I end up my talk and take any questions. These are all my mentors, and Balash is my direct boss now. He is a Nobel Prize committee member, and Mr. Harding is the one who is the possession of 1/3 of the radioligands in the world. And Sajaj Rada is the founder, he is the one mainly associated in developing the magnetic resonance image. And Professor David Thompson, he is my mentor again. He is a PET-CT inventor, and I want to thank my previous mentor, Sam Gabriel. Thank you so much.
So that was a very inspiring talk, I guess. So just to have some more questions. So is it nice? So I would like to ask the students, if not researchers, and if not at least the faculty, to have a couple of questions.
Thanks for the outstanding talk. And you spoke about artificial intelligence. Do you think in the near future we don't require physicians? Because these days I am reading lots of news coming up, like Google is ready to prescribe its users with its artificial intelligence.
No, actually, when you are a kid, you need some guidance, right? So without guidance, you cannot do that. The physicians are the guidance here. Definitely you need a guidance. You cannot play with. Because if something goes wrong, even the pulse rate goes down. So somebody should be having monitor, and you should know the compound which should be given. So that's why again I want to insist here. So we need not only a physician. So we need all the developers. So artificial intelligence, now computer is not everything. So you saw the Shankar movie, right? The emotion comes to the robot, but that doesn't mean that in a realistic way. So we have to be very careful, and artificial intelligence with the guided, it only will survive and take it to further. I don't think that without guidance we can.
Next part of the technical session is going to start. That is a oral presentation from the delegates. Oral presentation is total 26 facts. So it has been divided into 3 sessions. So the session 1 is this time. Afternoon there is another session. Tomorrow morning it will be a 3rd session. So oral presentation is for 3 sessions only. So 1 to 6 oral papers today, just now it will be presented. Poster presentation today evening only, 4 to 5. Those who are keep the posters, they can paste the posters in the lunch time itself or in the tea time. So referees and delegates will go around and see your posters. So postal presentation time is 3:30 or 3:45 to 5 o'clock. In BSc Zoology, and during the lunch time you just find out where the poster will be pasted. So accordingly you can post it. Then another announcement, awards. So poster presentation and oral presentation, individually 3 awards. One, first, second, third prizes are there. Oral presentation, 3 awards. First, second, third. Likewise, poster presentation also, first, second, third. 3 prizes. So referees and other delegates in the interactive and best work and best answer. So based on this, it is recommended the jury will take their job to select the award. So accordingly you proceed, and poster presentation, the time is very limited. You don't go on tell about the introduction, everything. So 7 minutes presentation, 1 minute discussion. 1 or 2 questions only, that discussion part, questions will write. So only 7 minutes presentation. Today we start with the 6th members.
I request HOD Professor M.P. Sanjeevan to honor Dr. Priscilla Suresh. I'm very much delighted to be here as a part of this international conferences. It was really wonderful. I have to thank Dr. Saravanan, Organizing Secretary for this international conference, and Dr. Arjunan sir, Bharatidasan University, for conducting a wonderful 2-day international conferences. So we are benefited by the invited lecture, talk, and now we'll start the session, oral presentation. Morning to all. Myself, RDP, doing research under the guidance of Dr. R. Harikumaran Nair, School of Biosciences, Mahatma Gandhi University, Kottayam. Title of my presentation is Bisphenol induces reactive oxygen species mediated toxicity in Chan liver cells. Bisphenol is an endocrine destructive compound mostly found in polycarbonate plastics and epoxy resins. And studies reveal that bisphenol may leach out from the polycarbonate plastics and epoxy resin into the environment. And several epidemiological studies also reveal the presence of bisphenol S in human serum, urine, and even in the umbilical cord. So recently, bisphenol is considered as an emerging environmental contaminant. And in this case, whenever an environmental contaminant or exogenous compound may enter into the human body, it will activate the xenobiotic mechanism and alter the xenobiotic mechanism and induce the production of free radicals. So here, we try to analyze whether bisphenol is a different concentration, whether it induces cellular toxicity by producing free radicals or not. Objective of my study to find out the role of reactive oxygen species mediated hepatotoxicity by bisphenol S. For this, we selected Chan liver cells. It's a hepatocytes, normal hepatocytes. The experiment design consisting of 7 groups. First group considered as normal control without any treatment, and second group considered as vehicle control, treated with 0.25% DMSO. Here DMSO used as a medium to dissolve bisphenol S. And from 3rd to 7th group, considered treatment group, were treated with different concentration of bisphenol S, such as 100 micromolar concentration of bisphenol S, 200, 300, 400 and 500 micromolar concentration. And after a treatment period of 48 hours, we analyzed different parameters such as oxidative stress parameters like reactive oxygen species production, antioxidant level, lipid peroxidation, LDH and cell viability assays. And methodology used in our study. After the bisphenol S exposure, the reactive oxygen species production were studied using Castibatiala et al. Method, and the SOD level studied using Kankar et al. Method, and the catalase and GSH level were measured using AP et al. And Elman's methods respectively. And the lipid peroxidation and LDH leakage also measured in our study using Bugent et al. And using agape diagnostic method. Finally, Metabolically active cells were studied using cell viability assays such as MTT assay using Mossman et al. Method. And after these experiments, the result obtained from these experiments were analyzed statistically using SPSS 16 software, IBM SPSS software. And following up the result obtained from our study. This image shows the reactive oxygen species production after the exposure of bisphenols. After 48 hours exposure of bisphenols. These reactive oxygen species are chemically highly reactive forms. And in the presence of reactive oxygen species, the DCFHDA style, that means dihydrofluorescein diacetate, is an image that after the DCFHDA fluorescent intensity staining method. And if ROS is present in the cell, the DCFHDA is a non-fluorescent dye. Dihydrofluorescein diacetate may convert into highly fluorescent dihydrofluorescein compound, and it can be read at 570 nanometer. And from this image, we can observe that the intensity found to be increased in response to increasing bisphenols concentration. And this intensity changes can be read at 570 nanometer using a multimode reader, and the obtained intensity variation depicted in this graph. And according to this result, whenever bisphenols concentration is higher, like 200, 300, 400, increasing with response to increasing concentration, reactive oxygen species production found to be increased. And from these statistical different level studied at 0.05 level using SPSS software. And when cells produce reactive oxygen species, these are closely related to antioxidant status. That's why we measured the level of antioxidants such as superoxide dismutase, catalase, and glutathione. The level found to be decreased in response to increasing concentration of bisphenols. That means, whenever the reactive oxygen species production found to be increased, the cell containing antioxidants are insufficient to scavenge the electrons, the reactive oxygen species are produced after bisphenols exposure. The activity of superoxide dismutase and catalase found to be decreased in response to increasing bisphenols concentration, and the activity of GSH level also found to be decreased in response to increasing bisphenols concentration. And whenever the reactive oxygen species production found to be high and the antioxidant level becomes less, in that conditions, reactive oxygen species may scavenge electrons from the lipid bilayer membrane, and that leads to lipid peroxidation. That's why we measured the level of. That's why we measured the level of MDA and LDH activity. This activity found to be increased in response to increasing bisphenols concentration. And we measured also the viability using MTT assay. Whenever the metabolically active cells, during MTT assay, the metabolically active cells can absorb the tetrasodium salt and can emit green fluorescence, and it can be read at 530 nanometer. And using this fluorescent intensity, we measured the cell viability using the equation ODF test divided by ODF control into 100, and the percentage of viability plotted in the x-axis. And the cell viability found to be decreased in response to the increasing bisphenols concentration. And from this cell viability assay, we try to measure the IC50 value of bisphenols. It's at 442.90 micromolar. And at this IC50 value, bisphenols was 50% inhibition of Chang liver cells. And these are the outcome from our study. Bisphenols may induce reactive oxygen species production, liquid peroxidation, and finally the reactive oxygen species may induce cellular cytotoxicity. And from this result, we can conclude that whenever bisphenols exposure is occurs, whenever concentration found to be higher, reactive oxygen species production is become higher, and it will decrease the antioxidant status and cause lipid peroxidation, and finally leads to the leakage of lactic dehydrogenase enzyme from the cell into medium due to lipid peroxidation. And so can we conclude that bisphenols may produce reactive oxygen species production and cause cellular toxicity. Thank you.
Are you trying to say that BPA is fine? And BPS is...
We also analyzed the BPA toxicity. Compared to bisphenol S, BPA is more toxic. IC50 value of BPA is 264.90.
But most of the drinking water bottles contains this BPS and BPA, right?
Yes, of course. But nowadays bisphenol A tried to replace to the bisphenol S, because bisphenol A found to be endocrine disrupting activity, mostly which affect in children and in women, which act as an endocrine disrupting compound, which mimic the estrogen activities. So bisphenol A tried to replace in polycarbonate plastic deoxyresins. From our lab, we also quantitatively analyzed the presence of BPA and bisphenol S. We got presence of bisphenol S more in thermal cache acid, especially in supermarket receipt. Concentration lies from lower LOD up to 22.2 microgram. That value lies in between this micromolar concentration.
Do we have any PET drinking water bottles in the market which is completely free of...
We checked the presence of BPA and bisphenol S from the mineral water bottle, but we didn't get...
No, I am asking, do we really have any water bottles in the market which is completely free of this BPS and BPA?
Yes.
Okay.
There are some bottles available with government regulations. You can find it in most of the high-end supermarkets. But local grade plastics are very contaminated with BPA.
Gathered here in the dais or behind the dais. Now I'm here to present my topic, regulation of hyperandrogenism by combined action of metformin and hyperglycemia in albino rats. You all very well know the PCOD. PCOD is nowadays a common syndrome in women. PCOD is nothing but polycystic ovarian syndrome, which was early called as a Stein-Leventhal syndrome after the person who first described in 1935 in an Italian scientist Antonio. They observed in a young married woman moderately observed and infertile with the two abnormal size of ovary, which like pigeon eggs. Then the hyperglycemia is a good medicinal plant to cure the PCOD. Its common name is Veliparuthi and Uttamani. Metformin is a hallmark allopathic medicine for PCOD, hyperandrogenism and diabetics. It's a early fault of hyperglycemia. My objective is to treat the PCOS condition using the allotherapeutic medicine metformin, to treat the PCOD condition using herbal medicine hyperglycemia along with the metformin. We chose the 24 virgin female rats. We separate the 4 groups, control and testosterone propionate and the metformin treated rats and metformin along with the hyperglycemia. Then the methodology, the following parameters are evaluated to test the hyperandrogenism. Biochemical parameters of LDL, HDL, cholesterol and triglycerides, and hormonal assays are FSH and LH, progesterone, estrogen and testosterone tests. And the results, this graph depicted the testosterone propionate injected group are reducing the HDL level, and the control metformin treated and metformin along with the plant treated group increasing the HDL level. And the next LDL is very much higher in other groups, and cholesterol also the higher rather than the control and metformin treated and metformin along with the plant treated group. And the triglyceride also the higher peak level in the testosterone propionate injected group. And the hormones are plays a major role in cause the PCOD. FSH is more higher peak in the testosterone propionate injected drug rather than the control metformin and metformin along with the plant treated drug. Estrogen also the higher peak in TP injected group. Progesterone level will be reduced rather than the control metformin and metformin along with the plant treated. Testosterone is the male hormone which is the more higher peak level in the testosterone propionate at the injected group. So I concluded my work. Metformin is a good healing medicine for PCOD condition. Another insulin sensitization and hyperandrogenism. At the same time, Puthanampatti is a good medicine for good herbal drug for PCOD and it enhances the immunity and it's also cure the PCOD. When this both drug metformin and Puthanampatti combine together to form a good impact rather than metformin alone. Thank you.
Do you know that metformin is the drug for mood disorder? Mood disorder. Actually, this can be treated for mood disorder, I think. Did you had some literature on it?
Please review only. I know that metformin's more impact in the PCOD disease.
Because it is mainly this drug was used only to treat mood disorder, psychosis.
Thank you.
Histopathological changes in pancreas of almoviscerate and dose-edible approach. Aspirin. Aspirin is nothing but is acetyl acid and its most famous cheap, easily available and also it acts as a non-steroidal anti-inflammatory drug. The aspirin has two main actions in our body. First one is it acts as an anti-prostaglandin, that is anti-inflammation and also fever reducing and also acts as a pain reliever. Then it also acts as it's a mode of action in anti-platelets, that is blood thinner. And not only for that, it also treats the headache, cold and any joint pains. Then the mechanism begins in inflammation. First, the inflammation is occurred by due to the COX genes. The COX genes is nothing but the COX1 and COX2. The COX1 and COX2 are derived from the arachidonic acid. The arachidonic acid is nothing but a secretion of a cell membrane. Then the COX1 genes is represent the prostaglandin and thromboxane. The prostaglandin is protect the gastrointestinal mucus and thromboxane is supports the platelet functions. And COX2 genes are also responsible for prostaglandins. It is the main mediator for the inflammation, pain and fever. But the aspirins, due to that, the aspirins inhibit the COX1 and COX2, though it acts as an anti-inflammation. Then recent literature says the aspirin not only inhibit the COX gene and also inhibit protein I-kappa B kinase beta, that is IKK beta, and that kinase is activate the nuclear factor kappa B, that is NF kappa B, and also the IKK beta is having the another pathway for insulin resistance by interfering with the insulin signaling. So hence we research more on that. To predict to observe the effect of anti-inflammatory drug of aspirin of various doses of young albino visceral rat. We are observing histopathological studies in the pancreas and also estimating the serum glucose level. Then hypothesis of my studies: the repeated administration of higher doses of aspirin could lead to the pancreatic damage. Then insulin resistance may also change the biochemical parameter. The objectives to analyze analyze the effect of various doses of aspirin in blood glucose level, hemoglobin and glycosylated hemoglobin, insulin and also the C-peptide, and also to detect glycogen and glycogen metabolizing enzyme. And another one to estimate the protein in normal and treated rat
and also examine the toxicity of aspirin in pancreas of normal and aspirin treated rat. Then materials and methods. The rat were divided into the 7 groups. First group is normal rat treated with the normal diet alone. The group 2 it's treated with the normal rat plus aspirins. The concentrations level is 25 kg by body weight. And group 3 is normal rat with aspirin concentrated 50 mg. And group 4 with normal rat with 75 mg of aspirin. And group 5 is normal rat with 100 mg of aspirin. And group 6 normal rat with 125 mg. And group 7 is normal rat with 150 mg. Then our studies, the methods will be followed for determining the blood glucose, the glycosylated hemoglobin protein, liver glycogen and glycogen metabolism enzymes is done with the help of biochemical parameters. And the level of the insulin and C-peptide is determined in plasma by the RA method, that is radioimmunoassay. And microscopic studies represent the sectioning of the pancreas, the islets of Langerhans. The result shows that fast result says about the fasting blood glucose level in normal rat and also the aspirin treated rat. First, the normal rat and the untreated rats determining the normally there is no level of increases, but compared with the treated one, here we can see that there is a gradual gradual increases in normal and treated aspirin. It's done in the 7th days and 15th days. The aspirins are treated with twice a day for 15 days. Then the effect of hemoglobin and glycosylated hemoglobin. Here the hemoglobin level will be decreases in treated rat compared with the normal rat, but at the same time the glycosylated hemoglobin level will be increases. Then the liver glycogen level. The liver glycogen level is also increases continuously. The liver glycogen level also decreases in normal compared with the aspirin treated. Then we detect the effect of plasma insulin and C-peptide level. This both insulin and C-peptide level also decreases compared with the normal one and aspirin treated. Then we affect various doses of aspirin on total protein. The total protein level also decreases in normal. The increases in protein level increases the percentage, but in aspirin treated it slowly decreases. Then glycogen metabolism enzymes. So this two color difference clearly shows that first the glycogen as synthesized, that is light blue colors increases in normal. So the glycogen phosphorus it's attained the same conditions, but in normal in treated with aspirin it differs. There is a changes.
You got 2 more minutes.
That photomicroscopic of an islet of Langerhans, and the A is represent the control rat morphology is it's at stake. But in G it's treated with aspirin, the morphology will be changed. The result concludes that the administration of aspirin progressively increased the blood sugar level and also altered many biochemical parameters. The cells of an islet of Langerhans of normal rat have round vesicular nuclei and pale pink cytoplasm. The normal rat contains the round and vesicular nuclei, but the cells of a diabetic, which leads to the diabetic tooth, the animal had a pyknotic nuclei. The conclusion: the aspirin is used as a therapeutic process for also the diabetic patients. We suggest the mild dose alone is safe. The future prospect, we have to predict the long term of chronic administrations are needed for dose determinations or aspirin treatment, and also investigate the molecular mechanism behind the insulin effect. Thank you.
Did you do some control experiments withdrawing the aspirin? Control experiment, because when you withdraw, no control, then control animals, how the pancreatic is the secretion is the same.
Control also we withdraw the pancreas like
No, what is the level? If the level is the same or?
No, sir, differ. Compared with aspirin, it's differ, sir.
So you were claiming that aspirin is really.
Yes, sir. Except India, some of the countries ban aspirin.
So can you do the experiment that you stop for a while and whether it can get back to the normal? For example, if you are conducting the experiment for a month, so if the effect is due to the the drug you are taking, aspirin, then you stop the aspirin for a while. Then did you do the experiment after 1 month? Is any improvement in the insulin secretion?
Yes, sir. For aspirin treated only, that inhibit the COX genes, the full mechanism will be inhibited, sir.
Right.
If you stop that aspirin, the mechanism, what can I say?
Recovered.
Yes, sir, recovery.
Because why I am asking this question, because this is the only drug has been given to all cardiac patients. If this is a case, I am with the client, even the pharma company will take this kind of research. That is my concern. So just a comment.
So my result is a high dose of aspirin only cause many damage. Depends upon the body weight.
But my clinical, my thing is, because clinical trials, they do everything. So whatever you have done, the same thing they might have done in the past. Because without doing this, they don't reach the drug in the market. So that means whatever research you are conducting, I don't know whether it add value to the community. That is my question. Thank you.
Better
This is the comment.
we go for allopathic, we use the plant extract.
Okay.
I wanted to check the novelty of the study. Okay, but anyway, this is an important issue because in India most people the treatment of aspirin, even to be honest, I am also working on aspirin in some aspects. Thank you.
Present about cardioprotective effect of stevia rebaudiana on male albino rats. My main focus on cardioprotective effect of stevia rebaudiana on male rats against myocardial infarction. First of all, what is myocardial infarction? Myocardial infarction is nothing but the rapid development of myocardial necrosis occurring as a critical imbalance between myocardial blood supply and coronary blood supply. Why it may be occur? Due to the formation of occlusion at the site of rupture of other manus plateau in coronary artery. There are three factors such as level of occlusion in coronary artery, as well as presence or absence of collateral circulation and time of length of occlusion are the causes for severity of myocardial infarction. Then acute myocardial infarction is leading cause of morbidity or mortality. Then risk factors like smoking, we also know smoking and a lack of exercise and also consumption of high fat diet to contribute risk, which are the causes for severity of myocardial infarction and other heart disease such as coronary heart disease. And the objectives of my present study is to analyze the phytochemicals of aqueous extract of stevia rebaudiana leaves through preliminary phytochemical screening, as well to evaluate cardioprotective effect of aqueous stevia rebaudiana leaves through in vivo studies. Then we go to materials and methods. First of all, we have to discuss about selected plant. My selected plant, as I said in objective, stevia rebaudiana. Stevia rebaudiana is one of 54 members of genus stevia. It is only one to that produce sweet stevia glycoside. Those plants can be used to do treatment of myocardial infarction. Then we go to selected animal model. My selected animal model is Radus norvegicus, that is Lister strain, which was weighed ranges between 160 to 200 grams. As well, for the purpose of acclimatization, it could be provide standard pellet feed and water and it's my experimental design. There are 4 groups. Group 1 treated as normal control, Group 2 served as experimental control, and Group 3 treated with the plant extract to normal rats, and Group 4 treated with plant extract to myocardial infarction induced rats. It is a pre-treatment.
Stevia aqueous extract was pre-treated to animals, then myocardial infarction was induced to animals for all groups except control group because it has a control by inducing isoproteinol 2 consecutive days with a dosage of 1 milligram per 100 gram of body weight of rat. At the end of the process, animals were sacrificed. Next, the blood were collected and serum was stored for the purpose of analysis of different parameters. There are 12 parameters to evaluate, estimated and analyzed for the further biochemical analysis. Those are the parameters like serum cholesterol, HDL cholesterol, triglycerides, LDH, SP, SGOT, SGPT, glutathioneine peroxidase, alkaline phosphatase, acid phosphatase activity, catalase activity and gamma glutamine transpeptidase. Results. My results showed through graphical representation. This graph expresses body weight and heart weight of the different groups of rats. As I said, group 1, 2, 3 and 4. Those graphs clearly show cholesterol and triglycerides levels of different rats. Group 2 had cholesterol very significantly increased cholesterol and triglycerides levels compared to group 1, 3 and 4. You can also see group 3 had slight increase of cholesterol level compared to group 1. As well as you can also see group 4 had significantly decreased cholesterol as well as triglycerides levels compared to group 2. Then HDL. HDL is specific because group 2 has a control group, experimental control because it's only induced by isoproteinol. It also has a myocardial infarction. It has decreased level of HDL. As well in LDL level is very higher level in group 2. SGOT, SGPT and LDH. Those 3 enzymes are referred to as cardiac markers. Cardiac marker enzymes. Those cardiac marker enzyme synthesis are also very increased level in group 2 compared to other groups. Not only that, we have also analyzed antioxidant enzymes like catalase and GPX. GPX is nothing but glutathioneine peroxidase. Those antioxidant enzymes are also decreased level in group 2 compared to other groups. The increase in group 4th animal having isoproteinol also induced and plant extract also pre-treated. So it clearly suggests the antioxidant activity have the plant extract. Finally, I conclude that the result collectively proclaims that the aqueous plant extract reduce myocardial damage by exerted by induction of isoproteinol, by lowering of cholesterol, triglycerides and LDL and cardiac marker enzymes such as SGOT, SGPT and evidently increasing the HDL level concentration. Thank you.
So, what is the number of animals used in each group?
For group we have 6.
But I didn't see any error diagram, error bar. So how can you show it? Because I'm thinking one animal data, which is not sufficient to conclude anything. Okay, please.
Any other question? I have one more question. What's the mechanism you would like to propose in lowering cholesterol metabolic action by your extract?
What would be the reason devices mechanism?
Reduce cholesterol level.
Cholesterol, high amount of cholesterol going to be heart function is very fast and it could be leads to high amount of cardiac output. So high cholesterol.
My direct question is how do that extract reduces cholesterol level?
I can't understand your question, sir.
Thank you very much. You finish this.
Good afternoon to all. My topic is a comparative analysis of antibacterial activity of biosynthesis silver nanoparticle and gymnemic fraction isolated from the acetyl leaf extract of Gymnema silvestre. Introduction. Nanotechnology deals with smaller particles about less than 10 nanometer. Nanotechnology is upcoming field with high scopes in medicinal technology. Many antibiotics for various pathogens are susceptible to various side effects. The phytochemical drugs can be used for overcome these side effects. So, the silver nanoparticles is synthesized from Gymnema silvestre. It is used as the drugs. My objectives: preparation of gymnemic mixture. It is characterized by GCMS analysis and in vitro antimicrobial susceptibility assay and biosynthesis of silver nanoparticles characterized by UV spectroscopy, scanning electron microscopy, energy dispersive spectroscopy, X-ray diffraction and FTIR spectroscopy. Materials and methods. The gymnemic mixture isolated from the acetyl leaves extract of the Gymnema silvestre and silver nanoparticles synthesized from the Gymnema same plant. We have to culture. I cultured the E. coli and Streptococcus aureus and antimicrobial evaluation by this diffusion method. The diffusion method used to do antimicrobial activity. MIC and MBC is used to for determine the zone of inhibition and time to kill assay is used to the antimicrobial activity and DNA fragmentation is what is the rate of DNA present in the pathogens and protein estimated by Lowry's method and it is also used for determine the pathogen cell death and total protein expressed by SPS page. The statistics analysis used by SPS. My results. This first one is aqueous leaf extract. Second one is silver nanoparticle extract. The brown color is indicate the silver particles. And in the peak is this graph is FTIR graph of the silver Gymnema silvestre plant atmosphere. What are the possible molecule presenting in the extract? The peak is indicated. And this picture shows is UV spectrograph of the Gymnema silvestre aqueous plant. And the silver particles is absorbed the UV light at the 600 to 620 nanometer. And this is the antibacterial activity of the Gymnema silvestre. This is the E. coli culture. This is the Escherichia coli culture. This is used for Gymnema silvestre. It is the Gymnema silvestre extract.
Can we help you? What's the problem?
This is the time killed to assay at the what concentration the cell growth is reduced. This is the used by biomolecules of the gymnast state. This is the total protein assay. This is the GCMS gas chromatography. Each peak is indicated the biomolecule present in the fraction. This is the DNA analysis. First one is control, second one is E. coli used by the genomic fraction. Second one is the streptococcus. Third one is the tetracycline used. This is the genomic fraction. This is the silver nanoparticles. First one is DNA is found that first. Then second and third, there is no DNA fragment, so there is no growth, and in the bacteria will go to death. My conclusion is when compared to silver nanoparticles, when compared between the silver nanoparticles and genomic fraction, silver nanoparticle is more effective.
Thank you. Due to shortage of time, we have time for only one question for this session. Okay, thank you.
Fine, I have done well. They have answered most of the questions. So I want to appreciate them. I would like to thank, outside I would like to thank the management of Nehru Memorial College for giving me an opportunity to chair this session. And I would like to thank our your head of the department of Nehru Memorial College, Department of Zoology, Shanti Madam, for her gracious presence, and as well as for the in this occasion she has invited me to chair the session. Thank you.
Pluripotent cell will form all the three germlets: ectoderm, endoderm, mesoderm, and the extraembryonal tissue, just like placenta. Pluripotent cells, they form only the ectoderm, endoderm, and the mesoderm, not the extraembryonal tissue. That's the difference. Now, multipotent cells, they form the cell type of a similar lineage. Now, for example, I am telling you bone marrow stem cells or blood stem cells. They form all the lineages of blood cell type. And then, then we have unipotent cell type, which is like, cartilages, bone, all the cells which will form only single cell type, which is unipotent. So, what's the stem cell? Stem cell, the classical property of stem cells that can divide and maintain its own progeny, so it will maintain a stem cell as well as it maintain a differentiated cell type. So, this is basically cell renewal. This is a differentiation paradigm. So, cell when it divides, usually a somatic cell will divide into two identical daughter cells, whereas stem cell will divide into a daughter cell and a differentiated cell. So that's basically your progenitor cell here, and differentiated further into major differentiated cell type. So the basis of stem cells, as I mentioned earlier, so that one is proliferation and differentiation. This is the classical property to differentiate a stem cell from a somatic cell. Somatic cell will only form two daughter cells which are identical. Stem cell will not form two daughter cells. It will form one daughter cell which is your proliferation, the other will be differentiated cell type. So this does include classification that I have done based on the source of stem cells. So as I mentioned earlier, the embryonic stem cells, they will come to blastocysts. You all can ask me questions either at the end of the talk or during the presentation itself. So blastocyst is the inner cell mass which we have in all the cells here. You can isolate these cells and culture them, so they make the embryonic stem cell culture. Then we have the adult stem cells, which is there in the bone marrow and all the muscle tissues. They will all have the adult stem cells. So these cells are basically there to maintain the homeostasis in our body. We have satellite cells in the muscles. So when we have wear and tear during the exercise and all that, so these satellite muscle cells, the satellite cells will differentiate to muscle cells and repair the damage that's happened. Then we have something called the induced pluripotent stem cell. So this concept started somewhere in 2006-07 by a Japanese group, where what they said is that we can differentiate the cell type. So what happens is that when we have a somatic cell, in our body all the cells are derived from a embryonic stem cell. So he said, whichever cell type you are having, we can make it back, go back in your evolution and make a primordial cell which is a stem cell. So he used 4 factors. So what he did, he took somatic cells, put in 4 transcription factors, and then he found an embryonic stem cell like cell type. So this concept started because when we derive embryonic stem cells from placenta, So we basically killed the embryo. And there were a lot of ethical, moral and financial issues that got quite a bit of controversy happened to use embryonic stem cell culture. So nowadays people have, there's a lot of restrictions still for using embryonic stem cells or to deriving embryonic stem cells because we have to use a lot of placenta, zygote gets lost. And so this came into picture where you don't need to have any zygote loss. You can take any skin cells and make it into stem cells. You can take liver cells, make it into stem cells. So looking at the conceptually how the therapy would work, so you can get the stem cells from the blastocysts, culture them in the lab. Then once the culture is over, you can differentiate them into whichever tissue you want, liver cells or muscle cells or neurons, and put it back in the patient. It was pretty simplified over here. But it is a very cumbersome process, and this process is very difficult to happen. Now, that was the adult stem, that was the embryonic stem cell that I mentioned. Now, what happens in adult stem cell therapy? So my topic will focus more on the adult stem cell therapy because this is allowed by our government, by the Indian Council of Medical Research, and we can do adult stem cell therapy in our hospitals for treating patients. You take the cell from the patient, you are taking particular cell type from the patient, try to differentiate them into a particular cell type which you want, and then put it back in the patient. This is the autologous transplantation. So you are taking a cell type from the patient, putting it back into the patient. Thereby you are, removing the concept of immune rejection and immunoreactions and all that. What I said was this was the guy in Yamanaka. So he is part of the concept of induced pluripotent stem cells. So you are inducing stem cell property in a somatic cell. So what he did, he took a skin fibroblasts, he put in the co-transcription factors, and then he was able to differentiate them into all different lineages. And then to put it back in the patient. Present is, I think, a couple of clinical trials that's ongoing with using IPCs in Japan. So one all stem cells have been used in the clinic. They have been used for all these different tissues. A lot of work has been done in skin replacement, in thalassemia is the most successful disease treatment that's been done with bone marrow transplants. And what we do in our hospital is on liver stem cell deficiency. So I'm going to talk a little bit more about this. This is our work. So I hope you all will be able to understand. So in the eye, when we look at transverse section of the eye, so this is the front part of the eye, and the back of the eye that you have. And you have got stem cells residing in different tissues, the eye, the limbus, the body, retina, and the conjunctiva. So this is the cornea. So this part is the cornea. Then you have the limbus in the proximity of the cornea and the conjunctival junction. And then you have the stem cells residing in different places. So conceptually, when we say that how stem cells are going to can be used for therapy in the eye, you can have embryonic stem cells, or stem cells, or iPSCs, or the induced pluripotent stem cells. Differentiate them into a progenitor cell type before transplanting into the eye. So how it really works? So when the cells get transplanted, they integrate and differentiate because we are not putting in the mature cells, we are putting a progenitor cell type. So they will integrate and differentiate. They can release parental factors which will allow the residing cells to repopulate again and regrow, and also to fuse with the existing cell type. So all these two mechanisms are a process together they work for when you look at the stem cell therapy. So in the cornea, what's happening? So cornea is the surface of the eye. So cornea basically having corneal epithelial cell types. So this is schematic representation. So you have epithelial cell types in normal cornea, and in this cornea you get scar formation. And then you take cells from different tissues, from different cell types, differentiate them, and put it in the normal cornea. And after transplantation, you get a healthy cornea. In the retina, this is more conceptually way. So you have normal retina, when you have a diseased retina with photoreceptors, the rods in the cones, you can again differentiate your cell types into specific rods and cones, and then transplant into the diseased retina. Most of this retina work is done in the mice experiments. So for clinical trial, I think is done only for a couple of diseases in the eye. So this is to show you that the posterior of the eye is the different cell types that have been used in the different animal models, experiment models for treating dental diseases. This is the back side, and this is the front of the eye where we have used different kinds of cell types also used to look at the corneal surface. This is to show you summarizing the different stem cells that have been found in the eye and the status they are in the different stages of clinical trial or animal experiments. So further up, few of my next slide will be on limbus, which is basically in the vicinity of cornea. And how we do the clinical application is pre-implantation, pre-transplantation is post-transplantation. The patient having a scar here and that gets restored to a normal eye after transplantation. So where is your limbus located? Limbus is an area that is in the brown area around the cornea. That's where the stem cells reside in the eye. So limbus is basically harboring the pigmented cells, and these pigmented cells are having stem cell properties that maintain the corneal clarity and the corneal regeneration. This is how the disease eye will look like. So when we have the limbus stem cell deficiency, the conjunctiva, that's the white part of the eye, that grows over the cornea and covers the cornea surface. So this is where you have the transparent cornea, and this is your schematic representation. We have the stem cells residing in this kind of box, box in the bottom part, and when needed, they will move up, differentiate and move to the surface of the cornea. So this is the limbus region that's drawn here. This is how the limbus looks like in the eye. These are the sections from our hospital. So if you look at, these are all the different areas that I was mentioning earlier. The cells reside in this kind of vicinity, and as when needed, they differentiate and move up to the cornea. And then you have here, in the peripheral cornea, there is limbus, it is a conjunctiva. So what happens is that when you have a normal hemostasis, you have the corneal cells that are here that get diluted, so they get reduced in the tears. Can I use the hand mic? Okay. Hello. Okay. So what happens in normal condition is that you have the cell cornea here. The cells are getting removed by the tears and rubbing of your eyes and all that. And the limbus cells, they migrate to the cell cornea. So this is just a type of error. The cell cornea is the peripheral cornea. This is the limbus, which has the stem cells, and this is the conjunctiva. And now what happens in this condition? Just click this one. Next one. Next. Yeah. Thank you. So what happens here is that if you look at this place, you have again cells moving out from the cell cornea. Cells also moving from the peripheral cornea. But since limbus cells are not there anymore, these cells are not getting replaced. So when the cells are not getting replaced, you will have a damage. That's why we call it as limbus stem cell deficiency. Next slide. Okay, so what are the conditions where you have limbus stem cell deficiency? When you have exposed to chemical radiation burns and all, you will have this kind of thing that happens in the eye. So what happens is the white part of the eye grows over the corneal surface and you can't see anymore. It is a very common cause in our society because we usually don't take up the eye so much when you are working in the farmers or working in the fields or in the factories people are working. So in a condition like that, what happens, we can use just the limbal area from the conjunctival and the oral mucus. That's the buccal mucus of the patient. Now, all this that I am talking about here is basically I am taking a tissue from the patient, growing it in the lab and then putting it back in the damaged place. So that's all autologous. So it is preventing all kind of immune reaction for the patient. So what is generally done here, you have a pre-operative patient which comes with this kind of condition. We do a liver transplant, then there is a suturing that's done, and after surgery, this is what the patient is able to see. Can I have this is a video, okay? Excuse me, can you just click this? Let's see if it works. Yeah. This is to show you that this is the last step where we are growing the cells, and then it's getting transplanted on the corneal surface. What is here, this page. You can see the surgeon taking the cells out from the amniotic membrane and putting it on the corneal surface. So, for doing this, what we found is that for all these different tissues, we had different labs using different protocols. So we found it a very difficult way to grow our cells. We could not know which is the best protocol for following for our cells in our hospital. So in that effort, we started looking at a method which can simplify the culturing method which can be used for culturing limbal, conjunctival, and oral mucus. So that we can have one protocol that can be used for culturing cells from all different cell types for a corneal transplantation, corneal surface transplantation. So what is the treatment modality? You get the cells, you get the tissue from the patient, you grow it in the lab on amniotic membrane, then you put it back in the patient. So the number of variability that was there in this whole process was quite a bit. So the processing of tissue was done different labs were using different methods. They were growing on different matrices, they were using different components. So that was a lot for us to deal with when we were working in a hospital and trying to look at low cost for our patients. So what we thought that we should have a protocol that is comparable, between that we can compare the outcomes in different centers as to how that transplant is working. It should be low cost and it should have clarity and it should not have any animal source. So we thought of simplifying the protocol. So what we did was that we modified certain components of the protocol and we found that we can grow the cultures using a modified ham septual combination. This is a medium that we use for culturing our cells. This combination can be modified and we can still get caudal cells from all the different tissues. So this is just to show you that when we have a unilateral damage, that's one eye gets affected, you take the cells from the normal. Now what is happening when you have both eyes affected? When you have both eyes affected, you are taking the cells from the buccal mucosa of the same patient. Buccal mucosa is inside the mouth. So you are taking the cells from the buccal mucosa, growing it into the corneal lineage and then transplanting it into the system. So this is just to show you how we do the work, so that tissue samples are coming from the theatre, from the operating theatre. So then we put it on amniotic membrane, grow them by chopping into small pieces, expand culture, then this is after transplantation. This is just to show you the cells when they grow, this is how they look like. They have small cubital shape, this is critical cell type. You have small size of cells, and conjugal cells are a little bit smaller, this become a little bit like mesenchymal cell types. This is just to show you the staining of the different markers that we can use here. So what we have used here, this is a corneal differentiation marker, this is a stem cell marker. You see the cells can differentiate into corneal lineage using all the different kind of tissue sources using our culture method. And this was the protocol that we showed in our paper, and this is the protocol which is existing in the literature. So what I'm trying to emphasize is that a small change in the computational ratio of two major components in the culture can change your total differentiation paradigm. Using our method, we have transplanted these many patients in our hospital. I think the recent number is around 50 odd patients, and we have success of around 90+%. And we have also done for the oral mucosa and the conjunctival, and we have did good percentage. We are trying to compile all this for sending it for publication. So far what I was telling you is that we take the cells, grow them in the lab, and put it back into the patient. So there was a component of lab that was necessary in this procedure. So one of the clinicians from Hyderabad, from Nehru Institute, he said that why don't we take the cells, directly put it on the damaged leg without culturing it? Will it grow? So he started doing this practice, and he found that on doing that, he was able to still restore the vision of the patients after the liver stem cell deficiency disease. So now at present, people are trying to compare the this method is for culturing the cells, and this is simple liver transplant. We're trying to look at the outcome and find out which is the better method. Then we started looking at, okay, why people are so much focusing on liver? Is there a reason? Is there a cell type specific property that liver is having, for which people are only preparing, and clinicians are preparing to grow liver transplant, only they are not using conjunctival and oral? We started looking at the markers. So when we looked at the markers, we found that the differentiation marker is a little bit higher in livers compared to the other cell types. Then we look at stem cell markers, also higher a little bit here. This is to show you that when we grow the cells in a culture and we try to look at stem cell markers, we see an increase of around 8%, which is much more higher compared to the other two cell types. This is just to show the staining as to how the stem cell cells are differentiated in corneal lineage. This is a connection marker. There was no increase apoptosis as done by the PK staining. So during all this process, we came across a literature which said that for all this differentiation and all, you need a particular signaling mechanism. And that signaling mechanism is called Notch signaling mechanism. It's a developmental regulatory signaling mechanism where it is present in all the cells of our body. It is highly expressed in a tumor. It allows the cells to proliferate. And that's one of its major functions. It also has a role in angiogenesis and all that. So what does Notch signaling do? It blocks differentiation and maintains stemness in all this kind of tissues, hair, bone, blood, solid tumors, and so on. Whereas Notch activates differentiation and maintains activates differentiation in skin and corneal surface. So skin and cornea, it is promoting differentiation, whereas in all other tissues, it is blocking differentiation. So what is Notch? So this is basically a transmembrane protein which is synthesized in the Golgi complex, then moves to Golgi, then moves to the cell surface, acts with ligand from another cell, gets a signal, then this signal releases a component of the protein which goes to the nucleus and acts as a transcription, allows for transcription. So these are the receptors of the signaling pathway. We have got 4 receptors and 5 ligands, and this is how the signaling will work. We'll move it a little bit faster. This is not so important. So, this is the intracellular domain. It moves to the nucleus and acts as a transcript factor. So we started, okay, let's look at notch. If it is really important for our culture, let's look at the notch signaling.
This notch, the differential activity of notch, that is detected is inducing differentiation in corneal cells. On the other hand, it's inducing
so notch has got a cell-type specific property. Okay. So in corneal epithelial cells and the skin epithelial cells, it will promote differentiation. Whereas in all other cells, it promotes stemness.
Why is there any reason?
They have not found the reason. I mean, I don't know the reason for that, how it is doing it. But I would think that it could be because of some specific promoter related to work with the notch. So it's actually the transcription factor. It's doing something on transcription and a promoter and all that. I would imagine something like that. I don't think something is much more superficial.
Okay.
Then we started looking at the signaling pathway across all days, and we saw that during day 10 of the differentiation, it's a potent experiment. So when we look at day 10, there is a decrease of the notch signaling molecules. And then we thought, okay, if the notch we are saying, it is maintaining differentiation and blocking of notch is going to maintain the proteinure population, maintain the stemness in corneal cells. So this is the endogenous label which we have here. So if you see here, this is a differentiated cell types, and you don't see the stem cell population here. So you don't see the proteinure population here. So we thought, okay, we start activating notch in different days and then blocking in the same way. So when we activate notch in different days, what we saw is that what would you expect? I would expect that since I'm activating notch, I would see more differentiated cells and less stem cells, right? So what we see here is that on activation of notch, we see more differentiated cells here. This is a differentiation mark, the corneal differentiation mark, and very less stem cell population. So we thought that okay, let's try to block it. So we started blocking different days during the culture. On blocking it, we found that differentiation also was blocked, and stem cell number was increased enormously. So this work now has a clinical relevance because it has been shown already that when you have the stem cell marker population, this is a direct correlation of the clinical outcome of the patient. More the stem cell population, the better the clinical outcome with respect to vision restoration. Lesser the clinical stem cell population, poorer is the visual outcome. So this we are at present kind of combining everything to send, get the manuscript written and sent out. This I showed you earlier. So these are all the different stem cell clinical trials that's going on in the retinal cell types. A number of companies also participated because this will help them to generate a lot of money. And stem cell synthesis is a progressive field, and people want to have monopoly on that to be able to control the field in the future for the clinical application. And eye has a better advantage of doing all this kind of work. The reason is that eye is totally image-based, organ. So any damage in the eye is done by visual experiment. You don't have to. There is no invasiveness here. That's the first point. Second point is that eye is kind of immune privilege because cornea knowledge is vast, so that gives a little bit immune privilege to the eye. And these are two major points for which eye has been there in the stem cell and the gene therapy kind of work. Much more faster work is done in this organ. These are different stem cell therapy that's going on across the world. Just since you all are students, I thought I'll tell you briefly a little bit about the career opportunities. There are a lot of stem cell institutes in our country. And some of them are really premier institutes like NSTEM and all, which are part of ISC. Then you've got NCPS, JNCS, we also do some stem cell work in our hospital. When the market analysis says that the worth of stem cell field is around $67-68 billion for the future history up to 2020. A lot of companies that they mentioned in Novartis, advanced technology and all that, they are investing a lot of money in finding out stem cell related work. So in order to find success, this partner like this for the reason that A clinician or a researcher or a pharma alone cannot cure a ailing patient. We need to have a mutual collaboration between all the key partners to help the poor man. Finally, I would like to thank our Chairman Dr. Bhujashiki, Dr. Rohit Shetty, I would like to present Dr. Kumar Manikavel and my colleagues and other members of the hospital. I would like to thank the funding agency of Nehru Foundation, DSG, DBT and NTC USA for funding our work. I think that's it that I have. Good, yeah, thank you.
So thank you, sir, for enriching us on stem cells and their application in treating the diseases, especially related to corneal stem cell therapy. I just have one question regarding this. What about any other treatment for glaucoma where there is loss of vision? Any trial has been gone on stem cells?
There is a trial that people, I think they are recruiting in Pune, there's a hospital, they are recruiting people for glaucoma. The problem with glaucoma is a neural disease. So the problem with glaucoma is that it is a connection that is there between from the eye to the brain, that's optic nerve connection. So sometimes what happens, because of the high pressure and all that, there's a damage in the optic nerve. If there's damage in the optic nerve, experimentally doing something is different and cerebrally doing something is very different. So still experimentally people are working on it, but I have not seen any success stories so far, because I have not come across.
Thank you, sir.
Thank you, sir. Thank you, ma'am.
Good evening. Sorry, good afternoon. Actually, my colleague, former colleague and my friend, my brother, Dr. Paul Murugan Ramasamy is not able to make it today because we requested him on online talk, but due to his clear commitments, he was not able to do it because it is midnight also for him now in the United States. So he given a title like Biomedical Diagnostic Application in Translation Research. I think everybody aware that in the morning most of the word I used was translational research and imaging. So now also I want to brief using my own expertise, and of course it is expertise of Dr. Paul Murugan also, because we called as twins, because when he was doing PhD, so I was a postdoc, and when he moved to UCLA, then I moved to Cornell, then he moved to Stanford. I used to move also, like in the more or less same time, and we were together, and he is there as associate professor now, and I am become deputy director in Nanyang Technological University. Pretty much same, we are doing the same research, but he is more in preclinical, but I am doing all preclinical to clinical also. And here, so I don't want to tell the story of what Paul Murugan is doing, because it is not good. Also on his behalf, I can tell something what exactly he is doing, but I know the research, but I want to tell, and the same title I want to explain what we are doing in NTU. And what here you are seeing is the campus. We have 3 campuses, and before that I want to tell the NTU has a medical school, which is Imperial College London and NTU joint medical school. That means the Imperial curriculum will be adopted and the degree will be given jointly by NTU and Imperial College London. The system of teaching is the team-based learning, which is a very, very new. Team-based learning means 6 medical students sit in one place, one table, and they discuss about the anatomy, for example. And they will, like the question and answer by themselves. The subject expert will be there, and all will be given iPad. So they will, highlight. It is called the learning studio. Any iPad, all the iPad will be connected to the learning studio, the projectors. So the particular person want to project their results, they can project and they can discuss with their subject expert. That is the idea. So which is working well, and we are the first batch, but first cohort is coming out this year. And here you are seeing, this is the main campus, what EMB, Experimental Medicine building. And this is another building which is called the Clinical Science building, which is Novena, 20-story building. So you can see all student, the learning studio, even the anatomy lab and all these things. And here on the 7th floor we have a cognitive neuroimaging center, which is basically human imaging. So we have MR, 8 different modality brain monitoring devices, starting from EEG to MR, MEG, magnetoencephalography. And here the translational, because when we talk the Paul Murugan title, translational, so we have a general research strategy for our own Think Translational Neuroscience program in LKC Medicine. And our goal is that early biomarker approach and early disease mechanisms and therapies. The thing is very simple. For example, other diseases you can control later stage, but brain diseases you cannot control. Once the disease caused, then that's it. You have to either retire and prolonging medication, and finally your life will be short. But for that reason, so if anything early biomarker approach, you can able to find the disease in early onset, which is very useful. But you cannot sample the brain and do the research. That is another problem in neuroscience. So for that reason, so we are looking for a peripheral biomarkers which we leak from the brain because of circulation. Again, there is a problem. I don't know how many of them know. We have a blood-brain barrier which allows only the selective things inside the brain. So that's why we are not affected by many of the viral diseases, right? So these are all the things we have to take into consideration and we have to work. So when you do this translational research, definitely we have to keep all the thumb rules and we have to work. And we have a very, very good team. Sorry. So the Balash is our, real, the captain of the team, and I am the co-captain. Maybe I have to run the facility and do the research. And these are all the team members. And we are working on the basic neurobiology, probe development, in vitro assays, preclinical imaging, clinical imaging. So that is translation. So I explained in the morning, translational means bench to bedside. So you have to start from the scratch and end up at the clinic. So that is a complete the platform we have, and we are conducting research. This is our team members. And the neuroimaging program at NTU, and we have 3 different centers. It is planned. So the first one is the preclinical. The preclinical imaging center mainly, we have animal models for any studies, including ocular. These animal models starting from mice to marmosets. So marmosets are the new old monkeys which lives with the siblings like a human being. It won't allow the other, like other macaque, they give birth and they will after separation they live separately. But these marmosets, new old monkeys, all live together as a sibling. And it is appropriate to conduct this kind of social, because the neuroscience is more related to social and cognitive. So that way it will be more appropriate. That's what we are having a scanner for all imaging modalities for this, because we are molecular imaging people. So we want to record the molecular events in vivo and static. And so the second one is the cognitive neuroimaging center. I already mentioned that there are 8 different modalities of brain monitoring devices we have. By the way, we can able to achieve and conclude what is the real disease and how it is progressing. We have a longitudinal solution. We have For example, one project called Helios, Health Living in Singapore. The idea is the first cohort people will come on 30 years and every 5 years the follow-up can be done. So that way how the aging is in, carried in Singapore, stress environment, how it is progressed. So this is one of the aspects. 10 million has been given to study the healthy living in Singapore. So that kind of project we are running, that is the one example. And another one is the Clinical Neuroimaging Center, where we have a 7 Tesla MRI, which is the ultra high field MRI. The thing is very simple. In the brain, if you have a micro bleedings, the microvasculature bleeding, you cannot able to make it the cause of the disease. So for that reason, the same patient will be subjected to 3T first, then there may be a question mark. The radiographer or radiologist may have some doubts, then we will put them under stress in 7 Tesla and we will scan and see there is a microvasculature bleeding or any other deformities or any lesion or something like that. So these are all our the thing. The idea here is that to support the concept of the Paul Morgan, that the diagnosis, especially to diagnostic purpose, the scan, the imaging is one of the non-invasive tool. Because the other things, if you want to do the thing, translation, you have to intervene. There is no intervention, but we do everything non-invasively, and that's the way we do that. And this is again how the center and what are the building and where in the basement we are going to put the machines, because 7 Tesla normally is heavy, 35 tons, the magnet, we cannot put it on the top. But now in the Community Neuroimaging Center, which is 15 tons, we put it in the 7th floor. So again, the area and other things, and these are all the animals, what you are seeing on the down, this is the marmosets, the New World monkeys. So we have all the animal model in place to do that. So to conclude, so the Cognitive Neuroimaging Center or whatever it is, basically we are conducting the translation image. So this is the center, how it looks like. And our projects are Cradle, Biobank, and the University Cambridge, and Arise. These are all the projects we are doing. And ongoing preclinical, these are all the scanners we have. And I already shown some of the scanner in my last talk. This is a preclinical scanner, which is changeable beds, and you can do mice and rat. So this is a PET MR, this is a PET CT. So that way you can get 3 different modality images. And this is the monkey scanner. So you can even scan the normal macaques in a sitting position to assess their brain capacity, cognitive impairment and other things. And these are all the things. This is auto radiography, which also, this is the preparation of the monkey. Now we will put it to the scanner. And this is the spectrum. And these are all some of the thing we are working with the different institutions in a translational science, especially NNI, National Neuroscience Institute, and LKC, our school, DSO, Defense Science Organization. So I think, there are probe developments, and I think we are mostly heavily working on radioactive, especially positron emitting radioisotopes, which will give the very precise result of what we are doing. We are synthesizing probes for metabolic disease and other things. This is one of the projects one of my PhD student is doing on how the leaky, the biomarker from the brain can be diagnosed. For example, the Alzheimer model was given. So that way he is doing, but we are considering blood, skin, and other things. And we got a very good result. We are thinking of communicating through nature, and we are submitting. So these are all the science we are doing. This is another one, how the traumatic brain injury, because the bike accident is most common in Singapore. So when they are biking, the rain always every week you can see 4 days of rain, 4 days in a week. So all the time the skid bike. Motorbikes, they fell down, and traumatic brain injury is more common. So, and also the government is very well supporting on the dramatic thing. So thank you. The translation of the whole bench. Thank you so much.
Because you mentioned about neuroimaging. First, in the human eye, there is so many neurovascular diseases out there. Do you did any
We are having OCT, ocular coherence tomography. So we thinking that the extension of the cornea, especially, is act as a threat. I think doctors have said. So because of that reason, see my, like especially he's a doctor and psychiatrist, my boss. So he cleverly put OCT. So that way, if you are seeing anything defect, then definitely it is related to some brain injury or some lesion or some defect in the part of the brain. So yes, we have that modality to cover that part. Yes.
Can biomarkers if we not to block those blood leakages?
Yeah, we are still in search of it. Yeah, if you have one, then we will validate yours for sure. Because we are also looking at it. Because see, it is not very easy. See, other organs is easy. You can done it very well. You cannot introduce unless otherwise to open the scalp, and which is risky in a way. So yes, we are also happy to collaborate if such real research question is there, and the thing is that you are most welcome. Funding is ours.
Thank you.
Funding is ours.
Good afternoon, everyone. I can see more girls here. At the end of the take-home message, I'm just going to give a small message which is very important for the girls. Okay? Okay. So I said that angels of sun, that vitamin D, we all know that sources from vitamin D, lights the vision of premature babies. So this is like a short story, and you can just follow it. I've made it very simple. So, okay, and here comes a car with a baby, which says that vitamin D may act as a preventive measure for the retinopathy of prematurity. This is the disease where I'm going to talk about. So later, and in this whole story, which it drives with the gears of 3 gears, where I will be talking about that vitamin D, the modulation in oxygen and VGF. So this whole story drives with these 3 molecules. So as we all know, this vitamin D is the source is from the sun, and it is vitamin D3 is metabolized from the skin and due to the sun. And it is metabolized by the next form. It is taken by the liver, dietary intake, and the final active form is from the kidney. That is called as a 1,25-dihydroxy vitamin D3. So you all know about the vitamin D functions. We all know that it is very good for bone, everything. But it is all the known facts, and it also have been known about in tumorigenesis, where they say that at the end stage, where the vascularization is there, that vitamin D can prevent the hypoxic condition of the tumor. So they say it has been known to inhibit proliferation of the cancer cells. It is known to inhibit angiogenesis, where I will be talking about in my further talks. And it also induces differentiation, where it reduces the tumor formation. So from this, you can also know that vitamin D is multifunctional. As you all know that it is good for bone, and it involves the absorption for calcium and phosphorus. And as I said earlier, it inhibits cell proliferation, it improves the immune function, and it is also it decreases the oxidative stress, the DNA damage. And the main one, it is new one about vitamin D is that it modulates angiogenesis, where I will be talking about the angiogenesis. So what is angiogenesis? It is new genesis, angio new. So it's like new formation of blood vessels, where you can see that where the new blood vessels are formed, the sprouts are formed from the pre-existing ones. So it is a natural process where the new blood vessels are formed from the pre-existing vessels. So, but this angiogenesis can be physiological as well as pathological. So, physiological means like where during embryonic development you need new blood vessel formations, and also during wound healing where the wounds has to be healed. And in eye also we have this physiological angiogenesis where in retina, that is the retinal angiogenesis, which is important for the vision. So there also we have the normal angiogenesis. But in pathological angiogenesis, what happens that you all see in cancers and also in eye, there are like major three diseases called retinopathy of prematurity, where it occurs in the premature babies, and age-related macular degeneration, which occurs in old age people, and also diabetic retinopathy, which is due to diabetes. So, and here this is the picture where you can see the normal vasculature. This is called a photograph of the retina where you can see the blood vessels, and here it is the like more blood vessels you can see, like more vascularized retina. This is the pathological retina. So there are many angiogenic factors known. So here I have listed few key angiogenic factors, but among all these key angiogenic factors, I will be talking about the vascular endothelial growth factor. It is called VEGF. Why? This is known as the main angiogenic factor to induce the angiogenesis, both physiological and as well as pathological. And this was first known as the vascular permeability factor, and it has like different types like A, B, C, D. And this VEGF, where it doesn't work is that when there is an imbalance, because when the imbalance is there in the VEGF, whether it is low or it is high, anything is low, anything is high, it will cause a disease, any factors. So it's same like the imbalance in the VEGF, it causes the pathological angiogenesis. So how does this VEGF acts? Actually it is involved, as I said, it stimulates angiogenesis. It promotes the chemotactic factors, and it induces the inflammatory cells, and it induces the pathological angiogenesis. So this main ingredients of angiogenesis, as I said, when this VEGF it is modulated when oxygen is low, that is hypoxic condition. When oxygen is low, this VEGF is high. When oxygen is high, this VEGF is low. So that the main ingredients, the oxygen can modulate the angiogenesis. Of course, the other factors are involved. So VEGF is involved with the modulating of the oxygen. So in this retinopathy of prematurity disease, we will be talking about the VEGF. So like vitamin D, then the angiogenesis. When the angiogenesis, the VEGF is the main key factor. And in that with VEGF, I'm going to talk about one of the eye conditions where retinopathy of prematurity. So this retinopathy of prematurity, as I said, it occurs in the premature babies, because as due to the premature, they are born earlier. Like I would say, they are like special kids, like all the fairies who want to see the world so soon. I would say like that only then. So these fairies are like kept in oxygen. So to develop for the development of the lungs. So this retinopathy of prematurity, India stands at the third level. So, and if you see the more darker blue, it is the more severity of the retinopathy of premature babies. And about 20% of the babies who are born premature are developing this ROP, retinopathy of prematurity. And the main key risk factors for the development of this ROP, that is the retinopathy of prematurity, in the preterm babies is that low gestational age. If they are born less than 32 weeks, you can suspect this ROP, and to severe condition if untreated. And due to low birth weight, and also due to the oxygen supplementation. And this here I am talking about the oxygen supplementation, though the low gestational age and low birth weight are there. In our experiments and in vitro studies, we have done with the oxygen supplementation where the mechanism is unknown. So this is a short video, as I said. It will be, this is the retina where You can see the blood vessel where it has like, before that I'll just briefly tell, it has two stages. As I said, it has an early stage where the blood vessel is not well developed because they are kept in oxygen, high oxygen temperature. And it is also a severe stage, the later stage where the oxygen goes down and the blood vessels are formed aggressively. Can you just click on it? So this is the retina. If you say that at the early stage, that there is a nasal retina, only the blood vessel has to grow to full, but it has like only half of the blood vessel is growing. And after that, because due to the high oxygen condition, the blood vessel is not able to grow. Then there is called stage 2, intermediate stage, where it is done, and stage 3, where the blood vessels are not properly formed and it peel off the retina if untreated, and the patients will become blind. So this is the disease condition of the baby. Next. So here I talk about now where we know that VGF is there and the vitamin D I have talked about, because there is preterm babies, like they are all born with systemically low vitamin D, like 95%, this is the report says that 95% of the babies are born with vitamin D. And as I said, they have two stages, the early stage and the late stage of the disease. Early stage where there is less angiogenesis, and the late stage where there is more aggressive blood vessel formation. So this we know that. And on the other hand, we know that vitamin D is deficient in these children, and we also know that vitamin D can modulate angiogenesis. And in one cancer study, like they have proved that at the molecular level, where that vitamin D acts upstream of VGF, and it can like induce the VGF secretion. So like vitamin D is upstream of VGF, and it can induce the promoter region, and it can modulate the angiogenesis. So here comes the question, like whether these patients, as they are born with vitamin D, and if we supplement them with vitamin D, whether they can modulate the angiogenesis or improve the retina. So as I said and as shown in the video, this is the early stage where the blood vessels are not fully formed in the retina, they are like half. And the stage 2, intermediate stage, here it is like hyperoxic condition, due to the high oxygen condition, and the VGF is less. This is what we imagined. This was the imagine, like we thought, so due to the less blood vessel formation, the growth factors or the VGF might be less. This is the second stage, transition stage from the hyperoxia to hypoxic condition. So but the current therapy is that the later stage they do like laser is just like sealing the leaking blood vessels. That is the current treatment ongoing. So in this condition, what we thought is that as vitamin D can modulate the angiogenesis, if we give them at the early stage so that it can induce the VGF. So whether this can act as a preventive therapy, as they are also having like less vitamin D. And later stage, due to this high VGF, vitamin D supplement, because this condition you can see it in cancer. In cancer they have shown that it is reducing the VGF. But here we doesn't know what is going to happen. So this later stage whether it can act as a curative therapy. But today I will be talking about the preventive therapy part, which we have also done some in vitro experiments in a laboratory. So first I will talk about as we know that first we were assuming that there is less blood vessel formation, so less VGF might be there. So for that we want to access the VGF levels in the patient samples. So but getting the sample from the premature babies is very critical. So the only source we can get is the tears of the premature babies. So from the tears, we just collect them with a strip of paper where just we wet the eye with the paper, and from that we extract the proteins from the strips, and we analyze the growth factors VGF. So and as I said, we collected from stage 1, stage 2 and stage 3 patients, and compared to the controls, these are normal babies who don't have ROP. And this stage 1 babies, the VGF is less compared to stage 2 and stage 3. So today I will be talking about the stage 1. And we also find that there is a correlation between vitamin D and VGF in the tears of these babies. So as you all remember, the main ingredients are called like the modulation of oxygen with the VGF, And so then we came across to see the concept like if we do add vitamin D with this modulation, what happens? So this comes in vitro experiments we have done, as you all seen the retina in the previous talk, Dr. Devish's talk. So in this retina is like very complicated layer. It's like 10 layers of retina is there, and it has like one main layer. It is called retinal pigment epithelial layer. Like one man army, I would say this is a single layer army. As a single layer, it plays multiple functions, and it is known as the blood retinal barrier. Like how Dr. He said like the blood brain barrier, it is the blood retinal barrier here. And so this is how we took these cells from the cadaver eyes and we cultured them. This is the RP cells where when you donate the eyes, after the anterior part which has been used for the donor, the remaining posterior region of the eye is used for research, where we collected this RP cells, retinal pigment epithelial cells, and as I said, it has like many functions. And the important thing is that in this RP cell function is not known with the angiogenesis, because in animal models, though it has been shown as it is lying at the back side of the eye, it is not visually. The damage is not visual. Though that in cornea you can see it at the front, as it is in the front, it is visual. So the importance of the RP due to angiogenesis, it is not given with the human diseases. So these are the results where I will be showing about in the RP cells, where we have seen that vitamin D, whether it goes down in a hyperoxic condition when compared to the normoxia, and we say here you can see that these are the vitamin D receptor and the converting enzymes, where they are high in gene expression. This shows that they are high in the normoxia when compared to hyperoxia. As well as the BGF and its receptors are also high in normoxia compared to the hyperoxic condition. When we supplemented them with vitamin D, you can see that when compared to normoxia, the vitamin D supplementation, it increased the BGF as well as the receptors levels of the gene expression, as well as also in hyperoxic condition. And then first we looked at the gene expression of BGF, then we wanted to see the secreted protein of the BGF, where we cultured them in hyperoxic condition and with vitamin D treatments. So you can see the secreted protein, which is the functional protein, is also it has been decreased in hyperoxic condition. And with the vitamin D supplements, you can see it improved in the hyperoxic condition. It improved the BGF secretion. And this is the immunostaining of the cells in for the BGF in the primary RP cells, where you can see the in hyperoxic condition the intensity is less, and with vitamin D treatments it is high. And this is the in vitro model for the blood vessel formation, where we have proved that functionally also that vitamin D can improve the blood vessel formation. This is like in normoxic condition how the blood vessels are formed. This is an in vitro model. And in hyperoxic condition, you can see there is no proper blood vessel formation. Like how I said, like it mimics the early stage of the retinopathy of prematurity patients. And here, these are the vitamin D treatments has improved the blood vessel formation. And it is also, this I am just showing the few results, like it has also improved the proliferation rate of the primary RP cells. So as I said that we are at the early stage now, that this is the bench to bedside translation part we are planning to do. So this at the stage 1 now we know that there is a correlation exists between VEGF and vitamin D. At the clinical side also we have seen, and also we have proved functionally with the blood vessel formation experiments that vitamin D can improve the blood vessel formation. And further we have to study about in stage 3 patients, and as well we have to develop the model for the stage 2 and stage 3 at the in vitro levels. So this vitamin D, as I said, this is my take home message, I would say, where you all know that apart from vitamin D, its function to bone, you all know that it is deficient in premature babies in India, and it can be, and this vitamin D can modulate the angiogenesis. And so this is where this whole story I've tried with all these 3 years in our B cells. And this deficiency is from where? Because it is from the mother. Because all girls, teenage girls, they are all with vitamin D deficient. There is also a report in India where all the especially teenage girls and who are going to be like mothers, they has to be like highly vitamin D rich persons. So that is what now government is also stressing, to have a program that women has to be enriched with vitamin D so that you can prevent that premature babies from RO. This is my message. Thank you.
Thank you for your nice presentation. Any questions from participant side?
Very good presentation. Nowadays, we are not exposed to sunlight. We prefer to stay in the AC room like that. And how will we get that vitamin D? In abroad and all, they are going for the diagnostic test in that vitamin D. Many of them are lacking due to various reasons because they are not exposed to sunlight. But here, women except girls and all, they should enrich with vitamin D. So what is lacking?
I would say that both, like as you said, outsource is not there, and intake. The second one is intake only. That is what they say recommend. So as you said, in Western countries, you see that sun exposure is lesser than us, but still that their diet is different, and also they are more health conscious, I would say, than us, and they go for regular checkup and they take intake of vitamin D. That is what our government is now stressing, as to have a program for this vitamin D, make it compulsory. This has to be this is not known. That is what
I would like to share. I read an article where the young students, they're using skin lotions, sunlight lotions, which is a very preventive measure, which prevents the synthesis of vitamin D. And the government has another program to fortify vitamin D in the milk. Yes, as they fortified iodine in salt, they have another program to fortify the vitamin D in milk. And I think it has not become successful because milk has not reached the entire population of India. That is why the statistic says. So whether it reached the deepest core of the population is a question mark. I think there's a discussion on it. But anyway, it is very thought-provoking, and current students should please take this message. All the lotions, cosmetics you are developing to treat you with diseases. You are the one causer for your disease. Yes, that's all. Thank you.
I have one more question. Which time is more suitable for the students?
Actually, vitamin D, this is general first thing I will say, they say that early morning is good, early morning sun is good, then it's not too much sun, afternoon 12 o'clock or 3 o'clock, that sun is not good, early morning sun I would say 6 to 8, that sun is good as per our country. So in Western it is different.
It depends upon the events.
Yeah, it is.
Scorching sun you avoid it. Even 5 to 6 or 5.
Thank you. Any question? No.
From Government College, and my topic is about analysis of salivary organic acid in buffalo with reference to the Eastern cycle. So as we all know that buffalo is the backbone of our Indian farmers, and their main economical importance is about their dairy and meat products. So when we compare with the cow and other animals, the eastern side in buffalo is a little bit dominant to the other animals. So in the related series, phenomenons of eastern cycle have been determined in the eastern cycle, and organic compounds like ascorbic acid, acetic acid, and propionic acid are identified in farm animals during the time of late estrus. And we well know that buffaloes are irregular breeders. They are not well breeders, so their production is less. So if you go for artificial insemination, even there's lack of efficiency of overpopulation, we cannot identify the estrus stage in the buffalo. So for the following factors, we are going to identify our aim is to identify the salivary organic acid, which is present in the different reproductive stages of the female buffalo through HPLC method. So by the animal selection is done when the animals in the estrous cycle, so it is detected during by their symptoms like cyanidation, hypersensitization like that. And the sample is collected. The sample is saliva through non-invasive method. And the sample is prepared through the solvent extraction method, and centrifuge is supernated through the silica 0.2 mu size membrane and done through HPLC to identify the organic acids. So this is the common basic graph of organic acid like ascorbic acid, acetic acid, propionic acid. The ascorbic acid is reduction time is 2.700, this one, and acetic acid is 3.273, and propionic acid is 4.663. So this is during the pre-stress time, the ascorbic acid range is 2.737 and acetic acid is 3.232. During the estrous time, the rate of the reduction time is 2.730 for ascorbic acid, and for acetic acid is 3.256. And during the time of diestrus, it is 2.732 for ascorbic and 3.283 for the diestrus. So when comparing to the overall stage, we show that the ascorbic acid rate is higher during the estrous stage. During the estrous stage, and acetic acid is lightly we can found it. So the overall amount, we indicate that ascorbic acid increases is 2.910 and during estrus is 4.251 and diestrus is 1.541. And acetic amount is just traceable amount we can find. So in overall, during the estrus, the rate is high. So here we conclude that the organic acid and here we consider organic acid like ascorbic and acetic acid concentration varies on their stages, whereas propionic acid is not at all present, it is totally absent. And the significant concentration between the ascorbic and acetic acid changes, where ascorbic rate is high during the estrus phase. And when compared with pre-estrus and diestrus, pre-estrus stage has the high amount of acid. So the major function of ascorbic acid is to enhance the corpus luteum and protect the hormones from not changing the pH. And in male, it is used for protecting the gametes and hormones. So as a result, we say that ascorbic acid can be used as an estrus detector. So with that detection, we can enhance their population. That's it.
I am Sania from Jammu College. I am going to present a general topic about which was entitled as extracellular polymeric substances from microalgae and their biotechnological potential. You know that microalgae, they are prokaryotic or eukaryotic organisms which grow rapidly and live in harsh conditions because they are unicellular and simple multicellular structures. For example, green algae chlorophyta, which has flagellum, combustion, pellicle, nucleus and chloroplast. And the signals, phenomenon mediated through various chemical signals which are facilitating the interaction and coordination of group of algal activities, and this is known as covalent sensing, and it often results in the formation of such physical structures with the unique characteristics known as biofilms. The biofilms are nothing but they are the sessile community of microbes which are irreversibly attached to the surface or each other, and which are embedded in a matrix of extracellular polymeric substances which are produced by those organisms. The extracellular polymeric substances are natural polymers which are of high molecular weight and are secreted by those microorganisms. And the EPS, extracellular polymeric substance, which established a functional and structural integrity of biofilms and is considered as a fundamental component to determine the physiochemical activities and properties of that biofilm. EPS, which consists of polysaccharides, and it provides a structure for those biofilms, and they are highly hydrated and bound to the underlying surfaces. These EPS may be associated with the metal ions, lipids, DNA and humic substances. The EPS production is known to be affected by the nutrient status of the growth medium. If the growth medium has excess available carbon, which promotes the EPS synthesis, and if the medium contains nitrogen, phosphate or potassium source, which decreases the EPS production. Extracellular polymeric substances formation, which consists of mainly 4 steps. These formation of biofilms are rather complex and it starts with conditioning film, second one absorption and attachment, third one growth and colonization, fourth one biofilm formation. First conditioning film. See, conditioning film is nothing but they alter the surface properties of the substratum and allowing the microorganisms to adhere on the surfaces. And second, absorption and attachment is still unknown, but the DLPO theory and the thermodynamic interactions are used to explain those attachment and absorption process. And growth and colonization is the production of polysaccharides that anchor the bacteria to surface, allow the colonies to grow. And finally, the biofilm formation, which is a fully developed biofilm which contains the EPS matrix and vertical structures separated by interstitial spaces, substratum, conditioning film, absorption, and then growth process. Finally, the biofilm formation, EPS matrix is there, and in between the vertical EPS structures, interstitial spaces is present. And these EPS are loosely attached to the cell wall or excreted into the environment. And many microalgae, which variety or red algae, cyanobacteria, or brown algae, etc., which are producers of structurally diverse EPS. These EPS are involved in the cell-to-cell interactions, adhesions, and biofilm formations. EPS are widely used in the food industry for thickeners and gelling additives, which improves the food quality and texture. Currently, the EPS have received much attention of their antibacterial, antioxidative, and anti-cancerous properties, which leads to the development of many pharmaceuticals. And types of EPS producing microalgae and its groups, bioactivities, and applications. The Porphyridium cruentum, which of group rhodophytes, which has antiviral, antibacterial activities, and Porphyridium species of group rhodophytes, which has intestinal morphological modifications, and microalgae of Rhodella reticulata of group rhodophytes, which has free radical scavenging antioxidant activity, and Anabenna spiroids of group cyanophytes, which has metal binding anti-thrombogenic activities. And finally, Nosta Klingia and Chlorella, which belongs to chlorophytes, which has metal binding, antiviral, antibacterial activity. And the main applications of this EPS are, they are most especially sustenance, animal food, biofuels, pharmaceuticals, and nutraceuticals sources. And in addition, these compounds of interest of microalgae, which excretes different extracellular polymeric substances. And then the biodiesel productions of biofuel productions are by EPS, and these algal biofilms are become increasingly popular as a strategy of conservation of microalgae, making the harvesting so cheaper. And another applications, they are used as an antiviral agent, anti-tumoric agent, antioxidants, anticoagulants, and hostile to inflammatories. They are valuable in mechanical engineering, for example, biolubricants and drag reducers. And in food science, they are used as thickeners and additives. Their properties are mainly due to their structural and physiochemical characteristics, which depends on the organisms which they are produced by. In the biomedical field, and polysaccharides from algae can be used to control drug delivery, wound management, and regenerative medicine.
Good evening. I am Sharini Ramke, doing research under the guidance of Dr. Sima Maheshwari of Peria VVR College, Tiruchirappalli. I wish to present a paper on surfactant-induced skeletal anomalies in Pulchis paralarva, a freshwater fish. Developmental stages in the life cycles of fish have been shown to be relatively sensitive to the various toxins. So, embryotoxicity tests are found to be relevant in assessing the contamination of the aquatic environment. Gross morphological abnormalities in the embryo, larval, and juvenile stages are sensitive indicators of exposure of toxins. Skeletal anomalies are found to be the major development abnormalities that affect the quality and quantity of the fishes, and also the axial malformations are mostly found that ranging from slight bending in the skeletal axis to the extreme of no axial development at all. It is documented that notochordal abnormalities in the early life stages have long-term impact in the form of deformed nervous system and vertebral anomalies. Literature survey reveals notochordal malformations in fishes on exposure to various toxins including heavy metals, petroleum compounds, pesticides and insecticides. But there is a lacuna in the literature regarding embryonic toxicity, embryonic skeletal development induced by surfactants. Hence, present study concentrates on sodium chloride sulfate, which is a widely used anionic surfactant, induces skeletal anomalies in the freshwater fish Purushes para. Sodium chloride sulfate is widely used because of its less cost and excellent forming properties, and it is widely applied in daily life products such as toothpaste, detergents, shampoos, shaving creams like. For this study, we have collected eggs from Pundia's para to induced breeding technique, and 50 fertilized eggs were introduced for the sodium chloride sulfate of different concentrations, 3 mg, 5 and 7 mg per liter, and developed larvae were photographed after 72 hours of post-hatching. We have observed skeletal anomalies like lordosis, which was the abnormal inward curvature of the notochord, then kyphosis, abnormal outward curvature of the notochord, and scoliosis, abnormal lateral or sideways curvature, and other tail deformities. Here I am showing the pictures. The first one is the normal one. The second one it is the larvae with lordosis. Then the third one scoliosis. The last one is the larvae with kyphosis. These are showing some tail deformities. In the normal larvae, the skeletal structure at the tail region will be pointed, but in this case some standard appearances are there. The second one is bending, the last one shows the bifurcated tail. This table shows the statistical support of these observations, that we can see that all the larvae exposed to different concentrations of SDS are showing abnormalities, but these results are found to be dose dependent. That means as the concentration of the toxicant increases, the percentage of abnormality also increases. The occurrence of skeletal abnormalities induced by surfactant in the present study is in line with the several authors who have worked with chlorpyrifos, peppermill effluent, pharmaceuticals like triclosan, diclofenac and carbamazepine, polycyclic aromatic hydrocarbons and malathion. The deformities of the notochord can be the result of controlled contractions of the body musculature which is taking place during hatching process, which may act directly onto the notochord, resulting in the bending or overlooping of the notochord, eventually results in the deformed nervous system also. This also suggested that the skeletal anomalies may be formed due to the inhibition of acetylcholinesterase, and also it is linked with depletion or deregulation of ions like calcium and phosphorus. So from the present study, we wish to conclude that surfactant containing detergents that are widely used in our daily life could induce embryonic malformations, especially skeletal anomalies in fishes, that may result in deterioration in the quality and quantity of the fish population. SDS at higher concentration could exert more severe damages to fish notochord that may be developed into vertebral anomalies that may persist during its adult stage also. So we are thinking that detergents are non-toxic or we can use it anywhere or in any quantity, but we should be aware of these as also have toxic effects. Thank you.
Good evening all. Thank you for this opportunity. I would like to present my work. Decellurized human amniotic membrane exhibits protective role under oxidative stress to maintain the cell functionality of the cultured ARP19. To begin with, amniotic membrane is part of the extra-embryonic layer. It's known as a since very old natural scaffold which has been used for many purposes, like especially for transplantation. This is obtained just after parturition, and extra-embryonic layer consists of amnion as well as chorion, but only amnion will be used for the culturing of the cells. And it consists of several layers: epithelial, basement, compact layer, fibroblast layer, all of them composed of collagen. And the basic property of amniotic membrane comprises its antioxidative nature, its anti-inflammatory nature, and its antimicrobial nature. And this is the structure, a healthy structure of the eye, comprising both anterior and posterior view. And the posterior portion of the eye, showing the microscopic anatomy of retina, has been displayed. And the main highlighted part is the retinal pigment epithelial cell, which possess many physiological properties like ion transport, barrier function, secretion of growth factors, especially some signaling molecules, so on. So in an oxidative stress-induced eye diseases, any dysfunction of this retinal pigment epithelial cells can lead to disease condition. So the disease condition prevails oxidative stress. So many types of diseases are like age-related macular degeneration, and the treatment modalities employed for this disease condition are using drugs as well as using cellular therapy. But all these treatment modalities are having its own limitations. So the current scope of this study determines how far an amniotic membrane and the ARP19 cells cultured on an amniotic membrane in an oxidative stress and in a healthy condition would help the cells to express more its physiological properties. So begin with, first we have to culture the cells on amniotic membrane. For that, the amniotic membrane has to be decellularized. The amniotic epithelial cells present on the membrane has to be removed through trypsinization, and on that decellularized membrane, the ARP19 cells will be cultured. And this picture shows the same images, and you can see that collagen fibrils around 50 micrometer in size. And after culturing ARP19 cells, the membrane on different culturing conditions, normoxia as well as in hyperoxia, has been aspirated for reactive oxygen species determination using immunofluorescence experiment. So the cells cultured on amniotic membrane under hyperoxia was showing the highest Ross staining, lowest Ross staining, but some tissue culture days under hyperoxia condition was showing the highest Ross condition, because it is already known that in hyperoxia condition the Ross can get elevated, but on amniotic membrane the Ross rate was completely generally low. And regarding the proliferation of the cells cultured on ham and TC was showing remarkable difference in terms of proliferation. Ham was showing both in normoxia as well as in hyperoxia least proliferative activity. So this would be helpful for in a disease scenario, like in a disease condition, the ARP cells under oxidative stress tends to proliferate more, and the cells, as it proliferates more, it can generate more aberrant cells. So in amniotic membrane, if you culture under oxidative stress also, the cell proliferation rate is comparatively low. Regarding the apoptosis, the cell viability as well as the metabolic activity are comparatively higher on the amniotic membrane. So amniotic membrane is supporting the cells in a very healthy way in an oxidative stress condition. So the FLIGA experiment has been done to determine the overall active caspase present in the cell. So that depicts on the amniotic membrane in hyperoxia condition, because caspase activity was very low. So amniotic membrane as a whole shows it's a strong indicator for the protective nature of the cells. Regarding the epithelial to mesenchymal transition, that since this is an epithelial cells, it should maintain its epithelial morphology even in the oxidative stress condition. So amniotic membrane was showing with very less stress fibers for the ARP19 cells. Regarding the VGF secretion, this is one of the crucial significant function of the ARP19 cells. In an hyperoxia condition, normally the VGF secretion will go, it gets elevated. But here, when we did an ELISA to know the secretory VGF level, that overall, that secreted VGF level was still comparable with the healthy condition in a tissue culture disk. So HAM is not generally making the cells to produce more VGF. Phagocytosis is another crucial property of exhibited by the ARP90. So in a hyperoxia condition, the phagocytosis usually gets elevated. But in HAM, in a disease condition, the phagocytosis usually gets reduced. But in HAM, even under the hyperoxia condition as well as in the normoxia condition, the phagocytosis was showing almost higher than the normal tissue cultured ARP90 cells. So the overall upshot of this study is, even in a diseased scenario, HAM functions as an excellent scaffold. This is what we require for a transplantation point of view. Like it is showing low ROS activity because HAM itself is having some antioxidative property which is not lost even in the oxidative stress. It was showing low proliferation. This condition would be helpful for age-related macular regeneration and retinopathy kind of disease scenarios. The HAM cultured cells are highly metabolically active and it shows low apoptosis, since this is a strong indicator for protecting the cells. It was showing low EMT in terms of less stress fibers, so it is maintaining the cells to remain in an epithelial nature. It is showing low level of secreted VGF, as well as the cells are able to maintain their phagocytosis at a peak by growing in and HAM.
First of all, I thanks to Honorary Secretary, Dr. G. Arjunan sir, the Saravanan sir, the K. Saravanan, and Ramesh sir, and also Rajesh sir. So I am introducing myself, Dr. G. Chandra, Assistant Professor in ABC College, Mayiladuthurai. So this is my topics: anti-cirrhotic residual effect of Escherichia popularea on female genital tract. So next. So many medicinal plants, particularly, are used by people in people in developing countries. Traditional medicine means nothing but here for folklore and village based medicines. Traditional medicine. So population explosion. Population explosion, nothing but the major population impact in China and India. And then for in case of developing country, women living poverty with access to low quality health services tend to have more children. Particularly example in African and Bangladesh. So the women health is very important one. So the developing country women, Slovenia, for African country women, they have more child and they have poor tended to health trend. And also they have suffer very anemic and some of the causative agents of developing country and village. The rural people of this developing country. Okay. For the contraceptive pills available in marketing. Contraceptive pills you already know about this. The contraceptive pill for women for fertility, anti-fertility effect pills available in marketing. These are the pills are more side effects because of the hormonal based pill and hormonal based bodies and non-plant and so many pills are available in marketing. These pills are more side effect. They have to be based on impact of very irreversible to psychosis. Then suppose the people have to be plan to further check, they have to be based on for the recent activity. Also irreversible menstrual cycles, they have to be based on this the contraceptive pills. So we can use it in this for traditional medicines purely on for health wise for good one. Okay. So that is we have to be plan to choose for herbal based medicine of for available medicine. Conventional medicine is more than available in all the region, forest region. So for example, for potential herbals available in dense forest, what happens? We cannot go for this, but difficult to forest collect the material. So available material, potential material, herbal medicine is available for anywhere. So we can easily collect and we have to be provide the make the preferred to medicinal properties. So that is my plan is. I took my plant is here. This Species Poplania, this common plant. For the plant is grow. This is my plant, Species Poplania. This is in Tamil Uvers. This plant are entire for these medicinal property, not only from particular flower and leaf, and the seeds and the flowers, the barks, bark. The tree bark also a medicinal property that is known as for anti-inflammatory and allergic and psoriasis, for medicinal use people for tribal and other villages. And then bark is for traditional medicine used by tribal people of Indra and the village communities. So we have took this study of this for anti-fertility and anti-sterility effect. So this is materials are, but we can took this material for model is a mice model, female mice model. So the normal saline for 0.9% for control one, another one for this duration for 16 days long duration of treatment. Another one is for withdrawal studies also the best effect for this herbal. Withdrawal means for anti-steroidal effect, and then the free from the 30 days. After that, the herbal plant eliminate the anti-steroidal effect. That is known as very good for medicinal properties of this species of organ. Another one is here OVX. The OVX is purpose for confirmation of anti-steroidal effect of plant. So it means we can remove of ovary from the mice. Then we have to be estradiol appropriate and with compound and both are from this mice. After that we have to be examined for anti-steroidal effect of the species of organisms. Okay. So all these experiments are carried out in the institutional clinical community. Okay, next one. These are there is no for hematological parameters for there is no variation when compared to these hematological parameters. For BC, WBC and frequency count, there is no variation from the hematological parameters compared to control one. So another one is also the biochemical parameters also there is no any variation when compared to this control one. Okay. So these are for significant reduce in this for serum estradiol level for compared to this control one for 16 days duration and 120 kg body weight and 250 mg kg body weight and 150 kg body weight. These are two are significantly decrease in the ovarian compared to this control one. Another one serum progesterone also serum progesterone on significance. So that the further what is known as here for 400 mg is known as here there is no any estrogen synthesis from this mice for 16 days duration, and also progesterone also significant decrease to this for 400 mg kg body weight. And then the withdrawal study they have to be they have to be so significantly increase the estrogen synthesis and the progesterone. Significantly, for example, for compared to this, significantly decrease in the 16 days for purely herbal treatment and significantly improve for estrogen synthesis for the control one. These are so conclusion. So the metabolic extent of the species of Poplania is considerable anti-steroidal effect in female mice. The anti-steroidal effect of the species of Poplania resulted in altered ovarian function such as accumulation of cholesterol, decreased level of 3-beta and 17-beta, and key enzymes of 3-beta and 17-beta activities, and thus the anti-synthetic effect, and also there is no any side effect from this liver and kidney, and also a safe medicine for anti-fertility and anti-serogenic effect, and also a withdrawal effect of this plant of this species of Poplininia. Thank you very much.
Thank you, sir. It's open for questions. Thanks, sir. I'd like to invite the next speaker.
And inhibitory effects of Corapita guardiances under in vitro condition. Kidney stone. The urinary tract system consists of kidneys, ureter, bladder and urethra. The process of urination begins in the kidneys. Kidney stones are hard solid rocks that form in the urinary tract. Kidney stones may also form in the ureter or the bladder. The medical term of the kidney stones, nephrolithiasis and urolithiasis. Types of kidney stones. Calcium oxalate. Calcium oxalate is divided into two types. Calcium oxalate monohydrate, calcium oxalate dihydrate, and then calcium phosphate, steroid, uric acid and cysteine. Calcium oxalate is the most common type of kidney stones. They tend to form when the urine is acidic. It means low pH. Another one. Calcium phosphate. Calcium phosphate is less common type of stones. They tend to form when the urine is alkaline. It means the pH, high pH. Steroid kidney stones are caused by the bacterial infections in the urinary tract system. Uric acid is the more likely to form the uric acid kidney stones. Cysteine is the most common type of the kidney stones. It is caused by the genetic disorder. I have chosen struvite crystals. Pathophysiology of urinary stone. Kidney stones contain calcium oxalate, phosphate, magnesium, uric acid, and the formation of urinary calculi involves the crystallization process, which includes the following: nucleation, growth, aggregation of crystals. Nucleation is nothing but is the initial process of crystal formation. Growth, the process of crystals increase in size. Aggregation of crystals, the crystals number in a cluster form. The stone formation may be either homogeneous or heterogeneous. Treatment of kidney stones. Extracorporeal shockwave lithotripsy. This method using shockwave to break up kidney stones and they are easy to remove. And then percutaneous nephrolithotomy. Percutaneous nephrolithotomy is the minimal invasive procedure to remove the stones from the kidneys. It is mostly remove kidney stone in the more than 2 cm in size. Ureteroscopic stone removal using small camera inserted into the urethra to remove the stones. Scientific classification of Corapita guarniensis, kingdom Plantae, order Helicus, family Lestiraceae, genus Corapita, and species Corapita guarniensis. Research outline. Urolithiasis, the process of formation of stone in the kidney and in the urinary tract. The use of herbal medicines is now widespread in the treatment of various diseases and disorders. Herbs presently available for the treatment of kidney stones or Corapita guarniensis. Medicinal properties of Corapita guarniensis. Corapita guarniensis is one of the type of medicinal plant which has antibacterial, antimicrobial, antifungal, anti-tumor, antioxidant activities, etc. Qualitative analysis. Presence of alkaloids, flavonoids, steroids, tannins, glycosides, etc. was observed. Objective: to grow urinary type struvite crystals in vitro and to evaluate this inhibition efficiency of methanol extract of leaves of Corapita guarniensis has been studied. The grown struvite crystals were characterized by FTIR techniques. Scope of the research: therefore, the purpose of this research will be provide a multidisciplinary approach in characterizing a major component of struvite crystals grown in vitro and in vivo to help formulate prevention strategies controlling urinary stone growth. This research is also focused to find the alternative herb Corapita guarniensis for the treatment of the struvite crystals in the urinary stone. Materials and methods: in vitro growth of urinary calculite. Struvite crystals were grown by single diffusion gel growth technique. Characterization of growth crystals to verify the structure and proper formation of growth struvite crystals from harvested crystals by FTIR. This is my results. A1 is the control one, B1 is with distilled water, C1 is 1% of the extract, D 2%, and E 3, F 4, and G is the 5% of extract. When compared to the other one, G is the higher concentration of added extract. The stone was gradually reduced. This is the harvested crystals. The measurements of the crystals. Statistical analysis for harvested Corapita guarniensis struvite crystals. This is the FTIR spectra of struvite crystal obtained from Corapita guarniensis, the gel method. Conclusion: with an increase in the concentration of Corapita guarniensis, the weight of the formed crystals was gradually reduced from 2.49 gram to 0.09 gram for the struvite crystals respectively. FTIR techniques confirmed its functional group of struvite crystals. One way ANOVA performed with treated and untreated crystal growth data obtained from struvite crystals showed significant differences. This study confirms that using Corapita guarniensis can promote the formation of ammonium magnesium phosphate hexahydrate crystals and reduce the nucleation rate of struvite crystals, a major component of a urinary stone. Thank you.
Thank you. Any questions?
I have one question regarding the FTR spectra. What is the FTR spectra to do with this here? Why is the FTR spectra used here?
FTIR?
Yes.
Find the functional group.
Have you set this functional group here? You are just showing the wave numbers, right?
Yes.
And definitely there are not going to be changes in the functional group. Only the size of the crystal varies here, right?
Nice.
So whenever you show your spectra, you just designate its, assign its functional group. That will be better.
Okay.
Right? Okay. And there's now what changes, especially the intensity of peaks or no peak appearance, no. Then it's a question whether it could be used as a diagnostic marker here, no? No? So next time when you use it, you assign the function group, it will be better.
Okay. Thank you.